it will be having a development of more biocompatibility they are using. So here also, if I'll tell about various type of application, in this case, like the cochlear implanted system means a biomedical device that restore the hearing for people with severe to perform a different type of sensorineural hearing loss by directly stimulating to the auditory nerve. So this type of biomaterial also used. Means if I'll tell about different type of key component and their biomaterial for the different type of sound processing external units, different type of we can capture and digitalize the sound. There are different type of biomaterials are there, like medical grade plastics are there, like polycarbonate blends, like lightweight and durable casting, direct different type of silicon rubber for soft ear hook and cable insulation. So different type of neural tissue regeneration also used. Like that, there are also using different type of titanium and magnetic alloy also for coupling with the internal implant. You can see there is a second image here. One is for the sound processor, which is a external unit, and which will be the main function of the sound processor, in which the medical grade polymeric or polycarbonate blends are used to capture and digitalize the sound. If I'll go to the implant, to the internal receiver, it will convert the digital signal to the electrical impulses. So what type of biomaterial can be used? Maybe the scientists are trying titanium, and it is also there. Titanium used for the sealing casing, like it is a corrosion resistant. It is like a biocompatible. Sometimes many scientists also tried for ceramics. Ceramic is having a good sealing effect and for insulation also. Like silicon elastomers also they are for used for the cushioning and insulation of the internal wiring. So if I'll tell about electrode array in this case, electrode array means first it will deliver the electrical impulses to the auditory nerve. So there are also different type of biomaterial being used, like platinum and platinum iridium alloy, useful for the electrode channel, which is conductive but biocompatible. And there are silicon rubbers are also there, which is flexible body of the array and easily inserted to the cochlear turns. And there are different type of other material also being used, like polyimide. It will be sometime used in the thin film electrode arrays for flexibility and miniaturization. So this is the case where we are using. So if I'll tell, there are other type of application, various applications are there in the case of orthopedic application, different type of nephrology application also there. So there are various type of nephrology application, different type of application is there in the biomaterial. So there are different type of drug delivery systems are there. There are different type of implantable device are there, which will useful for the tissue regeneration, and diagnostic tools also there. So it is also if I'll tell about mm, uh, it is also useful for the different type of nephrological application. If I'll tell nephrological application means it is used, this material also used to design and treatment and regeneration of the kidney tissue and related system. Means the material the material is interacted with biological system and different type of critical managing the renal failure, dialysis and kidney regeneration. So it will be helpful as a dialysis membrane and the hemodialysis membrane also. So if I tell, there are various type of biomaterial are used. For the common biomaterial are used for the filter waste from the blood in the patient with the kidney failure, if hemodialysis membrane are used. So there are various type of biomaterial also being used, like cellulose derivative biomaterial, like cellulose acetate, synthetic polymer, like polysulfolen, like polyether sulfolen, polyacryl nitrile. So these are the biomaterial being used. The properties, it is highly permeable and it is biocompatible, and it will reduce the clotting and immunoreaction. So in this case also there are different type of peritoneal dialysis biomaterials are there. Like catheter, we are telling about catheter. There are different type of medical grade silicon robots are there. Polyurethanes are there. Dialysate containing tubings are there. So there are different type of, but there are challenges also there. Means long-term biocompatibility and infection resistance. So sometimes there will be formation of a biofilm. So this also there are some constant also there. We have to take care about it. So there are different type of biomaterial also there for kidney tissue regeneration. Like for natural polymer we are using, means why for kidney tissue regeneration? Mainly for kidney tissue regeneration means we have to rebuild the damaged nephrons or entire kidney structure. So there are different type of natural polymers are there: collagen, gelatin, alginate, chitosan. Synthetic polymers are there: polylacticoglycolic acid, polyethylene glycol. So these are the there. So mainly what is the application are there for to revive the nephron structure, and also in the near future we can have a stem cell delivery and guided tissue regeneration also. Like that, for the drug delivery system, scientists are try for the nanoparticle and hydrogel for the renal tissue or control systemic toxicity. Renal compromised patient, they are using dendrimite based carrier, they are using chitosan or alginate micro nanogel, they are polyethylene glycosylated liposomes. So these are being used. So here in this case, so there are various type of, if you can see, just I want to see for different type of skin tissue regeneration also being used. Mainly, if I'll see, there is a pictograph of skin for any type of wound, chronic wound is there for, or diabetic foot ulcers will be there. So which will be different type of application can be there. So there are different type of hydrogels are there, different type of oleogels are there, different type of other variety type of application are there. So if I'll tell about, there are various application are there. So now just in a brief way I covered about orthopedic application. Initially, orthopedic application means there are various type of material are using for different type, like aluminum, zirconia. For dental application, zirconia, porcelain. For bone repair we are using also the biomaterial hydroxyapatite, bioglass. For cardiology, the scientists are more about the alumina, piezoceramic. For different type of surgery they are using zirconia, alumina. And for the drug delivery, different type of calcium phosphate, different bioglass are using. So this image just I want to share. There are various type of application are there, like epidermal application or dermis application. Means there are different type. If I'll tell about the skin tissue regeneration or different type of topical drug delivery application, so there are various type of biomaterial are using. And scientists are trying to make a gradient type of structure. In the gradient type of structure, there will be a biphasic material or biphasic scaffold type of material which can mimic the epidermal cell and dermis cell. But the thing is that if I'll tell about the skin tissue regeneration or any type of application, we have to also taken into account the if we are using any type of biomaterial, or if any type of infection are there. So whether there will be involvement of different type of WBC cells are there, neutrophil, eosinophil, basophil. Also we have to take care, is there any release of any superoxide or reactive oxygen species, or any inflammatory signal will come. So everything we have to taken into care. So if I'll tell, so the tissue engineering, there is a vast, if I'll tell, there are application, if I'll tell about the nano composite, I'm telling for different cancer and different type of skin, there are using various type of nano composite also application. So there are nano wires, nano rod, like if different type of electrons spinning materials are using. So if I'll tell about 2D structure, they are using various type of freeze drying method, frigilation, or they are using gas forming. They are using various type electrospinning. So they are using different type of nanostructure lipid carrier which will look like a cell membrane, hydrogel nanomicrosphere, polymeric nano capsules are there. There are different type of carbon nanotubes, carbon dots. and graphene sheets, hybrid nanocomposite, metal nanoparticle, and magnetic nanoparticle, which are these are the backbone now. So scientists, whomever working on it, so they want to make a nanostructure which will be, if they want to release some material. So if they want to release, let's suppose for we are using any type of plant extract, let's suppose that plant extract contain different type of flavonoids, tannins. So that nanocomposite material, some of the nanocomposite material they are using, which can also helpful in releasing the substance. So these are useful for the biological application, which is biocompatible, and which is having non-toxic and non-immunogenic, and there will be no rejection will be there. So nowadays there are various type of hydrogel also there. So there are various type of hydrogel being prepared from the natural polymer, like if I'll tell an example of hydrogel, there are two types of hydrogel are there. One is physical and chemical hydrogel. So best example I have given for the soft lens. So why the hydrogel also now in the market? Because hydrogel have vast application, not only for tissue regeneration application, for different type of food application, different type of food grade application, nutraceutical delivery application. So hydrogel, if you want to check a particular type of drug release pattern, how the drug will be released from this polymer, how the drug will be in that particular error matrix. And if I'll tell about any chemical hydrogel, means it is like a permanent hydrogel. It is particularly cross-linked with this some naturally polymer. Like if any type of two natural polymer are I am giving, I am adding some of the cross-linker. Cross-linker means anything. Like if I'll tell about glutaraldehyde, EDC, NHS, N-hydroxysuccinamide, genipine, glycogen. So it will make a different type of a cross-link type of a structure. So which will be useful for different type of application. So hydrogel is one of the very much important for different drug delivery to check. And for nowadays there are injectable hydrogels are there for different type of stem cell transplant carrier, for space filling scaffold carrier, for growth factor delivery into engineering application, different type of blood vessel, heart muscle application, different type of like skin application, nerve application. So nowadays it is having various type of application in the hydrogels are there. So if I'll tell about oleogel, one is hydrogel, which is made from different type of natural polymer. Let's suppose I'm telling any natural polymer. Generally, it is hydrophilic, gelatin. It can be, or it can be any type of polysaccharide, oligosaccharide, any polymer, or any type of protein. If I'll tell about oleogel now it is being useful for different type of lipid delivery system, for different ointment. Nowadays, like different type of oleogel also, like this formulation, they're adding some silver borate or different type of material for ointment purpose. It is like a, you can tell, semi-solid. Or nowadays oleogel having good market. It is also useful for intravaginal drug delivery. The ladies who are having lactating mother, or who cannot take that particular type of infection medicine, which can cause also some adverse effect to their body. Nowadays also it is being also used in this case of it. So there are different type of oleogel are made from different type of fat and oil. If I'll tell about different type of saturated or unsaturated fats, like different type of palmitic acid, stearic acid, oleic acid, heptadeconic acid. So various type of oil, fat, and different type of oil are using different type of oil, and we have to check in which oil and that fat proportion it is having a particular stable structure. So this is very much important, in which it will show some stable structure in this case of it. And in this particular oleogel it will having a stable structure, so that it can be used. We can add also, it is not for the drug, also we can use for the nutraceutical application. We can add some vitamin. Nowadays scientists are using various curcumin for this oleogel drug delivery system. Now if you can see, there is a particular type of a biogels are there. In this case of a biogel, they are using hydrogel plus a oleogel. Both the hydrogel, both the hydrophilic and hydrophobic, both the composition are simultaneously there for releasing of the hydrophilic and lipophilic compound. Nowadays, it is containing both the sandwich type of layer, both hydrophilic and lipophilic substance are there. So which will be useful for various type of drug delivery application. So it is useful for topical drug delivery application, generally for transdermal drug delivery application it is being used. For the, it is also used for the sustainable drug delivery, for the bioavailability as enhancement. So there are various type of organogels are also there, which will be for wound healing and tissue regeneration application. It is useful for the emol gel for the drug carrier control release, hydrophilic and lipophilic balance. So there are various type of dual drug delivery application for the targeted therapy, for customizable for patient specific needs. So it is being also it is being used. So if I'll tell, there are various type of different type of inorganic compounds are there, different ceramics are there. So nowadays, if I'll tell alumina, zirconium, calcium phosphate, silica, pyrolytic carbon, hydroxyapatite, bioceramic. So these are the different type of uh, materials are being there, so which will also useful for different type of bone tissue regeneration, cartilage tissue regeneration application. So you can see this is a different type of ceramics are using bioceramic. Nowadays they are using bioglass, which can also mimic the stimulated body fluid. Like various type of bioglasses are there, like sol gel based bioglasses, 1393 bioglasses, and different type of hydroxyapatite, tricalcium phosphate. It is inert to the body. It is having high wear resistance, and it is having a high modulus and compressive strength, and it is having a aesthetic property. Okay, so this is being used nowadays. Metal also, it will be used. Metal means, if I'll tell about biomaterial, the metal will play a vital role. So different type of metal also, there metal alloys, different type of implant. I told previously, for orthopedic application, for any type of application, we're using various type of for dental application also. Different type of metals are there. Metal tubing for different type of uterine drug delivery also, they are using various type of metallic implant also. So if I'll tell about next generation nanomaterial and biopolymer, so there are various type of nanoparticle, different type of nanomaterial, different type of nanosilicon hybrid based bio inks are there. And nowadays it is being useful for 3D bioprinting. 3D bioprinting play a vital role for the tissue regeneration, because we are preparing a bio ink. That particular bio ink is having a cell suspension, plus the biomaterials will be there. Both biomaterial and the cell suspension, it is being called as a bio ink. And there are different type of decellularized scaffolds are there. If I'll tell an example of decellularization means, if I'll tell about what is a tissue. Tissue is a group of cell combined with each other by extracellular matrix. So we are also getting the different type of decellularized scaffold for different type of animal. We are getting the decellularized, which is also that particular type of protein, which will be also useful for various type of tissue regeneration application. So here there will be a formulation of kidney derived cells. You are using different type of methacrylate based bio ink, and here it is being used. The means that means Mm. Means there are different type of are using different type of decellularization based method, being it is being used, like for the process of kidney, any adipose derived cell, many things. It is also one of the application of decellulose matrix also they are using. If I'll tell about biodegradable polymer, so just it is not that much new. Everyone knows about biodegradable polymer. Means biodegradable polymer means it explains the hydrolytic degradation of different functional group. Means biodegradable polymer means it may be focuses on carbonyl containing bonds breakdown in the presence of water like hydrolysis. So there are various type of hydrolysis and functional group with carbonyl bonded to the different type of heteroatoms. So means this shows the nucleophilic acyl substitute reaction where water attack to the carbonyl leading to the hydrolytic cleavage. So there are various groups are there. Ester, common in the polylactic acid, polycarbolactam, polyhydroxybutyrene. So amide bond will be there, mainly seen in the protein and polyamide. So another example I given thioester. Thioester also useful and relevant to the some biological active molecule also there. So different type of this biodegradable polymers are there. So here also if I'll tell about various type of biodegradable uh, polymers are there like previously I told you polyester, polyamides, polythioesters. So there are various type of imide hydrolysis will be there, anhydrated hydrolysis are there. So various type of groups are there. So means if I want to tell about this material, so there are various type of ester groups are there. That ester group may be contain a CO bond, which is like a polyester, like if it contain a amide bond, carbon and nitrogen like proteins, nylons like polyamides, like thioester, carbon and sulfur like various type of bioactive molecule polythioesters, anhydrides, different type of fast degrading drug carriers like imide, different type of 2 carbonyl on amide degrades to the acid and amide. So these are being used like hemiacetal, acetal. These are the biodegradable polymer being used. So nowadays, as I told, the biodegradable polymer are useful for various type of biomedical application also. Like if I'll tell, there are natural polymer, fibrin, collagen, chitosan, gelatin, hyaluronic acid. There are various type of polysaccharide. In the polysaccharide, homopolysaccharide, heteropolysaccharide. If I'll tell about homopolysaccharide, linear based homopolysaccharide, branched based homopolysaccharide. If I'll take an example of collagen, starch, there are different type of polymer, maybe decellulose matrix from the this can be also used synthetic polymer for enhancement of different type of strength like polylactic acid, polyglycolic acid, polycarbolactam, polytrimethylene carbonate. So this also nowadays the scientists are using. So if I'll tell about what are the factor which will be helpful in the hydrophilic polymer degradation. The hydrophilic more backbone, more hydrophilic end groove, more reactive hydrolytic groove in the backbone, less crystallinity, more porosity and smaller device like polyethylene glycol, polyvinyl alcohol, polyglutamic acid, polyvinyl pyrrolidone. So there are, you can see I had kept some images in which the functional groups will be there, which will be useful for, which will accelerate the polymer degradation. So we can check the polymer degradation by swelling study, by deformation, by bubbling, disappearance. Nowadays, also for food application. Now, if are using any type of food application, anything, so scientists are try to keep in the dehydrator. How much time the water will be dehydrated? But for degradation, we can check the swelling also. Like previous, we are using degradation by enzyme-based degradation to check the degradation. Like that, we are using, or we can also use thermal behavior like endothermic peak. Are there differential scaling calorimetry, different type of size exclusion chromatography, gel permeation chromatography, mass spectroscopy. So these are the use in the chemistry structure. If you want to check the change in the chemistry, so we can check the functional group by a different ATR mode of FTIR. We can check through nuclear magnetic resonance spectroscopy also. These are the thing can be used. So nowadays the scientists also try to use a biphasic scaffold. Biphasic scaffold means that is containing to like if this is like a in the biphysic of material of both the regeneration of bone and cartilage. This is like a example of a PLGA which will be enhance the osteogenesis and which will enhance the chondrogenesis. Maybe in the case of articular cartilage, if it will get in the tibia and femurs are there, if that articular cartilage get completely damaged, that articular cartilage glossy layer in which the articular cartilage in the knee joint, that is hyaline cartilage, which does not contain blood vascularization. There will be no blood vessels will be there, and it is a lymphatic and a neural system. So in this case of it, why I am telling, if any type of damage is there in the articular cartilage, which can also damage the calcified cartilage region and which can also damage the subchondral bone. So there we can also make a particular type of a biphasic scaffold in which it can enhance the osteogenic property and which can enhance the chondrogenic property. But we have to look into which material you are using and what is the strength. It has the ability to having the load bearing strength for the bone and both the cartilage, and we have to check the cell and cell interaction, whether there will be formation of a particular extracellular matrix or network type of structure, so that particular type of cell interaction is there or not. So we have to take care into it. So this is a paper long back I had given reference also, in they prepare a biphasic scaffold. Nowadays scientists are using like bacterial nanocellulose. So different type of bacteria they're using for nanocellulose, deriving nanocellulose from the bacteria, and it is useful for the biomedical application. Sometimes also it is having different type of role, and it is having some adverse also effect. But nowadays these are the main material they are using different type of thing. And these also they are using mycomaterial. If I'll tell about mycomaterial means, okay, you can tell it is like a fungal based material. Fungal based material means it will derive from a fungal mycelium. It is like a root like network, like in the filamentous of cell of the fungi, and they grow by feeding on lignocellulosic waste we can use. So it is also nowadays, it is also one type of biomaterial we can tell. Yeah, it is a renewable and it is biodegradable, and it is like a grown, we cannot manufacture, and naturally flame resistance and very lightweight. So it is having a good because it is a previously, everyone know it is having a packaging material. They are using various type of research paper, and they are using for eco-friendly alternative and different type of packaging material. Also it is useful for the different type of other like fashion material also using. But nowadays it will mainly the scientists are trying for a wound dressing material, this mycomaterial, because the scaffold with different type of antifungal and antibacterial property useful for this material. So there are various type of notable species are there, like in which that notable species, from there we can derive that one, like lucidum species, ostreata species. Like another also I told like bacterial nanocellulose, like the bacterial cellulose from xylinus species, like they are using from the bacteria. It is form a natural nanofiber during the fermentation, and it is like a highly pure. It is there will be in this case no lignin or hemicellulose, but it is having a nanoscale fiber which is higher in strength and flexibility, and which is hydrophilic, transparent, and biocompatible, and sterilizable also, which will be safe adaptable. So it will this mycomaterial and this material are mainly useful for drug delivery system, for wound dressing material, for moisture retentive and non-adherent. So there are various type of mycomaterial, as I told you, like Ganoderma species, like different fungal mycelium, different type of it contain different type of fibrous hyphal network structure, which is also having a good insulation and rigidity. For the bacterial nanocellulose, there are different type of, it will make a nano cellular mesh, which is higher tensile strength, which is higher flexible, and it will be useful for wound dressing material, and it is useful for implant and drug carriers. And generally it is bacterial nanocellulose. We have to take into consideration about two account. We have to do fermentation work also in the because it will be made from the fermentation. So these will be useful. So there are various type of nanomaterials are there. If means different class of nanomaterials are there for the tissue regeneration application. There are various type of nanomaterial, like we can tell gold nanoparticle. Yes, gold nanoparticle useful for different type of, it will promote the cell adhesion and proliferation. Useful for the biosensor application and drug delivery. If I'll tell about silver nanoparticle, everywhere we are using a silver nanoparticle. Because why we are using silver nanoparticle? Everyone in the bioceramic also, different type of scaffold also, they are using silver nanoparticle, because silver nanoparticle is having a strong antibacterial agent. And nowadays there are various type of cream, transdermal creams are there in the market. If you are having any type of infection in your body, like any type of herpes, or any type of acid fly, or any type of insect infection or there. So for the type of regeneration, we are using silver borate, also using various type of silver they are using, because silver having a higher strong antibacterial agent, and it is incorporated with wound dressing and scaffold also. And nowadays platinum nanoparticle also play a vital role. Different type of antioxidant and catalytic property potential for the neurogeneration, it is also being used now. And there are conductive polymers are there, like they are used to mimic the electrical property of the tissue, like nerve and muscles. And like polypyrrole is a conductive and biocompatible. Polypyrrole nowadays it is being useful for support the neural growth. And like polythiophene. Polythiophene is like a good electrical property and environmental stability, and it is also used in the muscle and nerve tissue scaffold. Like polyaniline, it will support the electrical simulation based the tissue regeneration. The bioceramics, as I told, calcium phosphate, it's the main mineral for the bone. It will promote the bone regeneration. These are the nanomaterial, nano hydroxy, like bioactive glass in the nano range. It will bond with the bone and soft tissue, replace the iron stimulating healing. Hydroxyapatite nowadays, a lot of hydroxyapatite using, because it is a natural occurring bone mineral. It is having helpful in the, not biocompatible, biocompatible everything, but it will be helpful in the osteointegration. If I'll tell about doped material, nowadays the scientists are trying to doped material. Why the doped material or metal ion are incorporated into the matrix? What is the need of this doped material? Doped hydroxyapatite, doped this one, bioactive glass. Because doped material, which is not, it will only enhance the bioactivity. If I'll tell about strontium, it will helpful in the bone formation. It will stimulate you, more bone formation will be there, reduced desorption will be there. If I'll take an example of zinc, it will enhance the enzyme activity and cell growth. Magnesium also promotes the cell adhesion. And manganese helpful in the bone mineralization. Now, and carbon nanotube, it will be known to everyone. It will be enhance the mechanical strength, and it will be useful for the neural and muscle tissue regeneration. Graphene support the stem cell proliferation. Now the scientists are trying for the using the stem cell for the graphene, and it is also useful for the neural and bone tissue regeneration. And if I'll tell about different type of nanoparticle formation are there, conductive polymers are there. So if are there, so how the nanoparticle formation being process being occur, means this diagram illustrate the nucleation and growth model of the nanoparticle, focusing on the that is called as a Lamar burst nucleation and Finke-Wadzik model. So this model explains how the nanoparticle formation will occur. So I don't want to get that much I'm telling. So but this is the main same case in which we can having, because these are the process are there, different type of super saturation, burst nucleation, and growth phase are there, like growth mechanisms are there in the case of Lamar burst nucleation, in which there will be a formation of the nanoparticle or multiple domain structure like crystalline or aggregated structure. There is also a called as a Finke-Wardzicki model, which will be helpful in the constant nucleation. Means nuclei like different type of green clusters are meant for the form the gradually and continuously over time, and there are different type of autocatalytic growths are there. Means different type of cluster catalyze further monomer reduction and particle growth, and these models fit the reaction where nanoparticle formation growth are slow and continuous. So there are various type of things are there. So if in a brief I am telling you what are those models are there, these models are generally, one is Labar model and is the Finke model. So it will take a complete thing to describe. Just I am telling a Lamar model means it will be a like instant model. It will be like growth mechanism means how it will separate from the nucleation, how the particle size being controlled through the narrow distribution, how it will be suitable for the rapid and controlled synthesis. So these are the things. So apart from this work, I try to go for the different type of cartilage tissue regeneration by using a different type of polymeric material, composite material, which is useful for different type of cartilage tissue regeneration. We try to make also some biophysics scaffold, which will helpful for the chondrogenic and osteogenic support, and we try to make the culture in a airlift bioreactor. So some bioreactor study also they are using different type of airlift bioreactor, rotating vessel bioreactor. So this bioreactor can be fitted to the CO2 incubator, and through peristatic pump we can give the air like airlift bioreactor. In a large scale, there are scientists try to prepare different type of chondrocytic or different type of cell. So now there will be a question for histopathological study also. So we have to check after the material we prepared for any type of cardiovascular, neural, osteochondral, chondral. We have to check whether there will be a what are the, is there any type of regenerated tissue is there or not, or whether there will be fibrosis will be there or not. So we have to check to different type of staining like Alcian blue or different type for glycogen content. For hemotoxin and eosin we have to check for the cell nucleus and extracellular matrix growth. So these are the study I being performed. So nowadays also there are development of various type of platelet rich plasma. So if I'll tell platelet rich plasma in the case of plasma is the main source, because if I'll tell plasma is the fluid part of the blood. In that plasma we have all the protein like albumin, immunoglobulin, which is the antibody. So platelet rich plasma and different type of polymeric material which can also like chitosan or chondroitin sulfate, which is a extracellular matrix of cartilage, that is also to make a particular type of a scaffold which will be helpful for the cartilage tissue regeneration. So here you can see there will be chitosan solution and there will be chondroitin sulfate, which is a extracellular matrix, and it will make a polyelectrolyte complex solution, and in which there will be addition of a concentrated silk fibroin. There are various type of sericulture farms are there. Silk fibroin is containing a various good mechanical strength. Nowadays for gelatin, like gelatin we are using for the everywhere, gelatin for any soft capsule, gelatin being used. Nowadays scientists try to make a silk fibroin material for different type of bone tissue regeneration application, wound healing, and cartilage application too. And as I told, there will be also different type of there will be biocomposite scaffold like polymethyl methacrylate, which is a composite scaffold for bone cement application. So there will be a bioactive glass can be prepared, and in that bioactive glass we can add we can prepare that composite scaffold by different type of leaching method. There will be a sodium chloride leaching method will be there. It will make a polymethyl methacrylamide scaffold, which we can add the silver nanoparticle into it for the as it is antibacterial, and we can check the calcination phase in which there will be a particular type of bioactive glass being formed, and that bioactive glass and the nanoparticle form a stable structure for bone cement application. So we try to make this paper being in the proofreading. Here also we try to make a particular type of a material, a particular type of a oleogel sample. Oleogel means like ointment, like a fat will be there, any type of fat, and it may be saturated, unsaturated fat. I told different type of oleic acid, palmitic acid, stearic acid, or any type of butter, cocoa butter, mango butter, or any type of oil into it. So we can add some drug into it, or we can add some surfactant into it. So we can make a particular type of solution which will be useful for different type of transdermal patch and transdermal application. So we have to check the control release in the topical drug delivery application. Topical drug delivery application we cannot check. We can check through different type of skin. We can take any skin, and there is a French diffusion cells are there. How there will be diffusion occur? There will be a particular type of diffusion. If I'll tell about oral application, we are taking any oral drug. In that we have to check through drug dissolution apparatus. But if I'll tell about diffusion, there will be transdermal patches will be there. So some of the also we try to prepare, while previously I worked in KLU, we try to prepare a particular type of film. Still the paper I have to communicate. So we have to high strength film for tissue regeneration, and that film is not at all brittle. So we have to make that particular type of a polymeric material. I don't want to reveal this material. So we have to take a particular material which have a particular load bearing strength, and that particular film is having a particular type of a transdermal patch we can use. So in the transdermal patch, we can check in the porcine model, and it is having a particular type of drug diffusion is also there. Scientists also try to make different type of ionophoric drug delivery system by giving some voltage and current to it. So there are various type of scale scaffold material. So nowadays, if I'll tell, I'll not take that much of time. Nowadays, there are using of 3D bioprinting. 3D printing are there, like stereolithography, fused diffusion molding, selective laser sintering. Nowadays, scientists are trying for additive manufacturing a 3D bioprinting. 3D bioprinting, they try to use the natural biomaterial like extracellular matrix they can derive, or any type of natural biomaterial which will be very much useful. And the cell, the autologous cell, we can use, or through stem cell also they are deriving, which will be useful for different type of tissue regeneration application. We can make a particular type of a model through CAD design by our own. So like there are different type of extrusion based 3D bioprintings are there, which can be also make a particular type of a bio ink. If I'll tell about bio ink, means it is like a biomaterial ink. Biomaterial ink means it will make a own your structure, additive manufacture. We are keeping a particular type of a biomaterial in which bio ink means already it is the cells are the main component, means the material plus cell. But in the 2D structure, we are using biomaterial ink. Biomaterials means you are preparing a particular type of a biomaterial scaffold in which that particular cell being seeded. So this is a pictograph representation. In this pictograph representation, there are different type of extrusion based bioprinting, inkjet based bioprintings are there, laser based bioprintings are there. So these are the bioprinting methods are using, where in which we are using the cells, we are using the polymer, and we are using the cross-linker to make a 3D bioprinted model. So there are various type of application for cancer research, for different type of transplantation and clinical application approach, different type of tissue engineering and regenerative medicine applications are there. So we have to make a biomaterial and cell, and we have already in the computer design, we are having a mesh design, and in which we can use a bioprinter. There are different type of multi-jet, inkjet bioprinter are available. There are in India also, there are various type of companies are there, like Cytolab, Next Big Innovation Lab. So there are various type of 3D bioprinter companies are there. So nowadays it is also the future of this particular type biomaterial. If I go to the 4D bioprinting, there may be safe memory polymers or safe memory alloys are there, but this is the best material bioprinter are available, which can be useful for different type of tissue regeneration application. So bioink, it may be biocompatible, it have a good injectable property, it should having the we have to check various type of rheological parameter. We have to check the cell compatibility, cell distribution, cell activity in a particular type of a sterile environment. We have to prepare this bio ink. So these are the different type of natural polymer, and different type of flow properties are there. Which will be over, and which will be advantage over the 2D bioprinting. 2D printing means 2D printing, sorry. So we are using, but 3D bioprinting play a vital role. Nowadays they're using different extracellular matrix, natural polymer, alginate, collagen, gelatin, chitosan. There are various type of polysaccharide. Like if I'll take about chitosan, different chitosan derived also they are using. Chitosan means it is an acetyl glucosamine and acetyl galactosamine. They're using chitin also. They're using fibrin, silk fibrin, agarose. Agarose it is a heteropolysaccharide. It is a protein. Gelatin is a protein. So every time they're using for different type of vascular application, ligament regeneration, tissue specific application, it's being used. So different type of myocardial tissue application, chronic wound application, different type of cancer model, articular cartilage application, these are being used. So sometime we are using some primary cell, somewhere using some cell line, and different type of stem cell also one of the source we are using, and we have to add some growth factor also, some like cell signaling molecule for different like transforming growth factor, interleukin 6, one morphogenic protein 2. So sometime we have to use connective tissue growth factor. These also being used in this case of it. Generally it is also used for different type of tissue regeneration application, tissue restoration application, and extracellular matrix generation application it is being used. So it is being the 3D bioprinting is useful for different disease model, different tissue engineering application, organ development, drug development testing, and personalized medicine, different type of cancerous research also it's being used. So as I briefed it out, so there are various, I'll not take this amount of time. In the scientist, this is the first case study in they prepare through a vascular model and tissue construct. There will be endothelial cell lines will be there. They're using different type of cell to, they are adding some transforming, some growth factor are used for the blood vascularization will be there. And there are different type of for cutaneous wound defect. They are using alginate containing the bio ink, which will and later they have taken the fibroblast cell, and the later they through the hematoxylin eosin strain they check that particular type of regenerative tissues are there. And there are also different type of multi-layer mineralized constructs are there, which will be useful for chondral and osteochondral tissue regeneration, like a gradient type of scaffold structure. It is being used. Then there are different type of neural tissue regeneration for spinal cord injury. It is very much difficult spinal cord injury. There will be spinal cord L2 bones. So there will be some compress in the bones will be there. So nowadays also neural cell and different type of bio inks are there in this case of it. So if I'll make a brief conclusion, so there are also in this future era of biomaterial, there are different type of bioprinter application are there, 3D bioprinting application, which is also one of my major interest in the research area. So I'll also applying for a couple of project also in this area, which will be very much useful, and which we can also design according to that particular material pore size, and we have to and which will ensure the biocompatibility, because we are not using any type of non-toxic material, and it will having a good tunable mechanical property, and it is having a good advance in the 3D bioprinting, and it will be helpful in the different type of functional organ model and different type of cancer research. So this is the just bioprinting. We have to take a design, so it will be like it will be in the CAD design we have to design. So this is like it will be a particular type of a additive manufacturing technique. So in the additive manufacturing we can prepare this one. So I will not take that much of time. So nowadays also as a faculty development program, so there are here also we are using different type of artificial intelligence in the education, like one gamma apps are there. So if it is in a very rush or very hurry, if you want to prepare any PPT, just you can give the title of the talk. So in that particular Gamma app, so it can generate the PPT according to your interest. There are different type of puzzle apps also there, which can also generate different type of quiz, different type of crossword puzzle also there, and different type of quiz networks are there, and different type of AI studios are there, which can make a particular video, short video, and quiz application and PPT are there. So these are the content generation tool and collaboration tool. So thank you very much. So I try to make it within 1 hour to complete. So thank you very much for giving me this opportunity. I hope I'm little bit fast because I want to cover all this biomaterial approaches. So I try to cover some of the some might be I left out, but some brief I had covered the biomaterial and its application. So and it is a very good platform. So in the near future, also I can do some work into it like 3D bioprinting model. So I can also share my research outcome also. So now also I am in the verge of developing the lab. So thank you very much. Uh, thank you. My voice is audible. Hello.
Yeah, sir, your voice is audible.
So thank you. I had done with my presentation, Dr. Somnath Roy, sir. So
Thank you, sir, for this wonderful session. So we learned a lot about this biomaterials and bacterial nanocellulose and many other things. That was a great addition to my knowledge. So now I would request the participants to come up with their questions. So the house is open for question answers now. I see lots of appreciation messages coming in the chat box. Divya
They
says
are
last
asking you to
suggestion.
share the PPT.
Yeah, there is one suggestion to like, yeah,
uh,
participants are asking if it is possible to share the PPT for their.
so actually, I will share the PPT with Dr. Ubnes, Dr. Gaurav. I have their mail ID. So I will delete some of the slide which are the research work I communicated, and 4 5 slide. After that, I will share this PPT. Around 6 7 slide I will delete, and after that I will share with Dr. Ubnes or Dr. Gaurav. Or no issue at
Yeah,
all.
perfect. So yeah, the house is open for discussion. If somebody is having any questions, please come up with your questions. Uh, I have a quick question for you, sir.
Please,
Maybe it's
please.
a very naive question. So I was just learning about this bacterial nanocellulose. That was an addition to my knowledge. So my question is, are there any, like you mentioned that nowadays to prepare this nanocellulose, you are using fermentation, etc. I mean, typically fermentation things.
Uh,
Is it possible to introduce, or I do not know if there are any reports where I was quickly checking that Acetobacter is probably one of the bacteria which is producing this nanocellulose. So
yes, yes,
my
yes.
question goes in this direction. So is it possible to bring this recombinant DNA technology approaches or metabolic engineering to improve the yield of the cellulose so that like if we are thinking in a larger scale, so like is it a scalable thing or it's still in the laboratory uh, uh,
No,
conditions?
no,
I'm
no.
making all.
Actually, the thing is that Yes, bacterial nanocellulose through this acidobacter strain can enhance through recombinant DNA technology because
Okay.
these species are it can be improve the yield and property. But the thing, if I'll tell about it is like a if I'll tell the recombinant technology and surely Dr. Somnath. So I did not have that particular type of thing. So we have to like insulin, we have to check the recombinant DNA technology enhancement. We have to check the gene expression, and we have to check the overexpression of cellulose synthesis also. Different type of knockout, different type of we have to check. But I did not find, only they are using in some of the paper I find it out for particular type of bacterial nanocellulose preparation they are using different sec plus method and to
Oh,
make the enhancement. But the main aim that they did not use that particular type of nanocellulose, in that they mix some of the thing, because it is a natural polymeric material. It is like
mhm.
a like it is a homopolymer of beta 1 for linked
Yeah.
glycose. So I don't it may be the recombinant DNA technology may be enhanced, but there may be some pro and cons will be there. uh, so as per
So
my
I mean, in this, okay, so for this, I mean, that was my question. So this for production of nanocellulose using this bacterial method, so far nobody has tried this recombinant DNA technology interventions, right? If I understand
Yes,
correctly.
actually, I did not gone through it, but
Okay.
recombinant DNA technology can be a particular type of enhanced, which can improve the bacterial nanocellulose production. So we have to go for different type of experiment, Dr. Somnath sir. We have to take for different type of pathway, different type of introducing different type of genes for to check its property, and we have to check the increasing the transcription rate. So we have to go for the genetic engineering, molecular biology, fermentation, all the application we have to take. So it may be a costly application, but we can do. Maybe this recombinant DNA technology, maybe it is being there, but I did not go through any
Okay.
paper into it. So might be I will look into it also once.
Okay, sir. Yeah, thank you, sir. There, I see a question coming from Dr. H.S. Rathor. He asks, is microbiomaterials, I mean, do these materials cause any pathogenic reaction, pathogenicity?
Which
Probably
mycomaterial?
he asks about this mycobiomaterials. I think he mentioning from the biomaterials coming from fungi, maybe. That's what his
Yes, 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 yes.
question is in direction. If they are pathogenic, that's what he's asking.
No, 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 no. Means that we are deriving from that mycelium, which is a vegetative part of the fungus. It is not considered as a pathogenic, but some fungi that produce mycelium can be pathogenic to the animal. But we have to take a particular species. That mycelium itself is not typically airborne. It will not. No, no, no. That particular type of we have to take a species. So which is not this thing. So we have to,
Non-pathogenic.
yes, yes, yes.
So, Dr. Rathor, would you like to follow up on the question? He adds, Dr. Rathor, you can just unmute yourself and follow up if you want. Uh, he has added something for human use. I mean, I think he wants to ask that whether these I mean, this biomaterials coming from this fungal origin are safe for humans. Probably that's what he want to ask.
Uh,
Acha, he is unable to unmute. Let me check.
No, he can, all participants can unmute themselves.
Unmute, yes, that's what yeah. Dr. Rathod, you can unmute yourself. It's not an issue.
Is it audible, sir?
yes, yes, yes.
Yes.
Good morning, sir.
Good morning.
Yeah. Actually, I wanted to know that is it any study is there for biomaterial as you inform in your PPT? They are going to use for human use, so it may cause something later for any means not compatible or any reactive kind of thing, any immune rejection or anything like that.
No, sir. Actually, it will not that much use. But I had studied. There are various type of fungal or different type of, they are using, they are deriving different type of carbohydrate like beta glucan, and it will be used. Sometime it is really useful the carbohydrate source also, and some mycomaterial also it will be safe and effective. We have to go to the particular type of I did not do the work, but mycomaterial papers are there. We have to like different type of Ganoderma lucidum species or different type of Pluterius ostreus. It is having a promising biocompatible and regenerative medicine application. Like that, we have to take this material. What we are want to derive? We want to derive any type of carbohydrate. We want to derive any type of material. So it is safe use for. So I saw some of the paper. It is safely useful for dermal substitute.
It is an up to animal study, sir, or it is
So
far
it,
more for
no. If it is safe to use, we can go through. You can go through in vitro study. Later, we can also go for the animal model study also. We
can I
can.
ask one more question, sir?
No, the thing is that it is like a if I'll tell about, it is like a uh, self-growing, if you can add in a biomaterial scaffold. There are various type of literature are there. So if you can tell like 3D bioprinting also, three-dimensional printing mycelium hydrogel for different complex material, they are using that particular type of myocomposite for different type of structural biomaterial. So that means like bacterial nanocellulose, they are deriving something which will be useful for tissue regeneration and which is not and toxic. And mycelia from like edible fungi, like different type of species they can use. So it can be used, it will be not that much harmful, and it is suitable for the different type of dermal tissue regeneration also.
Sorry to disturb, but we are getting late actually. Next speaker is waiting outside.
Sure.
So we can
So,
have you just write and mail to us.
okay, so
Please go ahead
thank
with the
you.
next speaker.
Yeah, just a second. Thank you, Dr. Sharda Prasanna Malik, for your wonderful presentation. So, please join me in presenting the e-memento to sir. Just a second. Sir, can you kindly stop sharing your slides?
Yes, yes. Stop share, stop share.
Yeah.
I guess, sir.
So, thank you once again, Dr. Malik, for your wonderful presentation. And I would request Dr. Yuvanesh to kindly hand over the e-memento to sir.
But thanks a lot for inviting me. Sir, Shada sir, please kindly accept the email on behalf of this FDP.
Thank you very much, and thank you the team. And I am very much thankful to that Golgotia University and MNIT Allahabad Institution Innovation Council also for providing this opportunity to me. And many participants are also there. More than 300 more participants are there, and it is I hopefully it will be fruitful for the all the participants. And thank you very much. Okay.
Thank you, sir.
So
Thank you.
hello, Vijay sir. I will also listen to your session. I am there for one.
We are also waiting to listen to
Vijay
his
sir, can you
lecture.
recognize me?
Nice to nice to meet you, sir. Actually, I unable to listen your presentation completely. I will just miss your presentation.
टूटा तो नहीं है
Know the
Kind,
तो नहीं गिरा
1 second, sir. Sir, 1 second, sir.
जी
All the participants kindly mute yourself because we could not able to hear to the speaker's voice. Somebody is singing.
Participants, you are requested kindly to mute yourselves, please, so that the session can be smoothly conducted. Yeah, looks fine now. Okay. Okay, so moving further with our next speaker. So please join me virtually to welcome our next speaker, Dr. Vijay Kumar, MR, he is working as an assistant professor in the Department of Pharmaceutical Sciences, School of Pharmaceutical Sciences, Baba Saheb Bhimrao Ambedkar University. So, sir, welcome on board for this today's development program. So, before I hand over the stage to you, please let me take the opportunity of introducing you in front of our audience today. So, Dr. Vijay Kumar did his PhD from the Department of Pharmaceutical Engineering and Technology, IIT BHU, in 2016. He did his graduation, post graduation degree in pharmacy from the Tamil Nadu Dr. MGR Medical University, Chennai, in the years 2007 and 2010, respectively. He supervised one PhD and 26 M.Pharm students. Presently, 3 students are registered under his supervision, in addition to 7 M.Pharm students for their dissertation work. He has co-authored 22 research and review papers in peer-reviewed international and national journals. He has one granted patent to his credit till date. He has also presented 9 papers as author in various national and international conferences, and he has so far published 4 book chapters. He presented his research in multiple research presentations in international conferences at London, United Kingdom, Paris, France, etc. He has 3 extramural research grants and one institutional grant of rupees 85 lakhs. He also delivered 3 invited talks in different universities. He is currently working in the area of targeted nanomedicine and microneedles. He is a highly motivated teacher working towards technology transfer of his research output. So I take the opportunity of welcoming Dr. Vijay Kumar once again to our faculty development program. Welcome, sir, and the stage is yours.
Thank you, sir. Thank you so much for the kind introduction. Am I audible to everyone?
Yeah, yes, sir, you're audible.
Yes. Fine. My screen is visible for all of you. My screen is shared already.
It's not still shared.
Okay, let me check it. Is it shared now?
It has, yeah, now it's fine. It's in the slide mode, so you can shift to the full screen mode, sir. Yeah, perfect.
Very good morning for each and every teachers joined over here. Actually, I received the information, Dr. Yunesh, regarding this invited talk before 3 days. Within the short period of time, I could able to manage the slides which I usually used to perform the research work in my laboratory with some basic introduction about the transdermal drug delivery system. As the title says, I am going to talk about microneedles. But microneedles, if I am starting directly, it may not be highly relevant in drug delivery area. So I want to first talk about something on transdermal drug delivery system. What is the meaning of transdermal drug delivery system? How it is works? What is the difference between the topical drug delivery system and the transdermal drug delivery system? And then we can go ahead with the actual talk on microneedles. As we know, the delivery of drugs through skin is a historical and long time research. In that, there is a basic difference between the delivery of drugs to the skin or through the skin. That means whether we are going to deliver the drug exactly to act on the skin to treat some local diseases like fungal infections or cancer or any other diseases, or we are going to deliver the drug across the skin to treat some systemic diseases. Like if the drug is not going to act on local area, it will diffuse through skin and it will go somewhere else in the body and it will exert its pharmacological or therapeutic action. So I want to have some discussion on transdermal drug delivery system first, and then we can move ahead with the actual microneedle topic. The basic difference between the topical and the transdermal delivery system, whenever we are applying some ointment or paste or cream or gel on the skin, is it crossing the skin and moving towards the systemic circulation, and is it being circulated throughout the body? The question is still answerable. And transdermal tubular system, as we know, that transdermal patches are available in the market. It is being applied on the skin, and the drug is believed to be transferred through the skin and reaches the systemic circulation. And it is useful for the treatment of several diseases. It's well proven. And why the dermal applications like ointment, paste, gel, cream are not moving inside the systemic circulation, and why the transdermal patches are moving, why the drug molecules are traveling from the transdermal patches to the systemic circulation continuously to treat the disease. First, we have to clarify on this part why the ointment are not giving the transdermal drug system, but transdermal patches are expected to have the transdermal drug delivery system. While we are applying the dermal or topical application of dermal or topical ointments, in that case, what happens? The drug molecule will penetrate through, will not penetrate through the dermal part. What happens when we are applying the ointment, paste, gel, cream on it? A small quantity or small number of molecules are being applied on the skin, and when it is left as such, it will be dry, and the actual the drug reservoir or actual drug availability on the surface of the skin might be depleted, and because of that, the drug molecule could not penetrate inside the skin. It will be a local action on the dermal part. Instead, in case of transdermal drug delivery system, it is not going to dry, and the drug reservoir is going to exist on the surface of the skin for a prolonged period of the time, so that the continuous passage of or passive diffusion of the drug molecule from the transdermal drug delivery system to the systemic circulation will be continued, and the drug delivery also will be continued. So this is a we can say it's a very basic information and difference between the dermal and transdermal drug delivery system. And when we are going for the topical application, as we see in the second figure, if you could able to see the cursor movement, you can see that when we are applying on the topical cream, it used to adhere only on the skin surface. It will not, the drug molecule will not penetrate inside the skin. It will not penetrate through the skin to reach the systemic circulation. But in case of transdermal drug delivery patch, the drug molecules are moving ahead. If we want to deliver the drug inside the human body, that means to the systemic circulation, another way is a injection through a hypodermic needle. We can go for an injection formulation through a hypodermic needle. When we are injecting the drug through hypodermic needle, it will reach the systemic circulation, and the drug molecule will travel throughout the body, and it will give the pharmacological or therapeutic action systemically. The problem is when we are injecting the drug molecules through hypodermic needle, it will be painful, and patient incompatibility may be there. And apart from that, it require a trained person, a doctor or nurse should be there to administer the drug through the skin. When we are look out the microneedle patch, this is the actual interest of talk today. It is having the advantage over all other applications, like in comparison to the topical cream, it will not deliver the drug only onto the skin. It can also deliver the drug through the skin to the systemic circulation. Microneedle patches can be useful for the topical delivery, that means for local skin applications, or for the transdermal systems, like systemic delivery of drug molecule can also be achieved by using the microneedle patch. Over the transdermal patches, when we are applying for the transdermal patches, the quantity of drug moving from the transdermal patch to the systemic circulation is highly limited, because the presence of stratum corneum and epidermis and these layers are absolutely blocking the drug to transverse through it, and reaching the drug molecules to the systemic circulation. The skin barrier will prevent the number of molecules reaching the systemic circulation. And in case of microneedle, the number of molecules reaching the systemic circulation will be very high because it evade the stratum corneum barrier. That means the topical barrier, the outermost barrier of the skin will be overcome by the microneedle. It will be pierced, and the drug will be delivered directly to the dermis spot, so that the drug molecule movement from the patch will be very high in case of microneedle patches. Compared to the hypodermic needle, when it is painful, when we are applying the drug using the hypodermic needle, microneedle patches will not give any painful injections, and the biomedical waste being created from the hypodermic needle won't be existing in case of microneedle patch, and it will not painful, and it will not require any other person or any trained person to apply the drug molecule or apply the patch to the human skin. And moreover, the cost of microneedle patch is production of cost of production for microneedle patch will be lesser in compared to the hypodermic needle or injection or sterile formulations. So with this advantages, the microneedle patches are trying to get its space in the market. As of now, no microneedle patches are in the market. They are at the phase 3 clinical level for some small molecule delivery as well as for some vaccine delivery. No need to unpack. No need to unpack. With this, let me move ahead with some more basics about the transdermal regulatory system. Transdermal patches are very simple patches. It used to be available in 5 into 5 centimeter patches, or maybe a bigger size of patches also available. Just we have to peel the covering layer, and then we have to keep it on the skin for a prolonged period of time. For example, some patch may be active up to 1 day, or it may be 48 hours patch, or the patch may deliver the drug for 1 week. There are some patches, exceptional patches, deliver the drug molecule until 1 month. 1 month patches also available in the market. And the transdermal system having several advantages over the injection or oral drug delivery system, like it can bypass the pre-systemic metabolism. That means the liver metabolism while we are taking the drug orally will be overcome by the transdermal patches, and when the patches are being applied on the skin, it will deliver the drug for a prolonged period of the time, so that we do not worry about forgetting the dose or taking more number of doses, or frequency of doses is not required. Very less dosage frequency may require, and we can terminate the treatment at any time. It's not like a permanent injection given inside the human body or depot injection. Whenever we want to just discontinue the therapy, just we need to remove the patch and the treatment will be discontinued, and it release the drug content for a prolonged period of the time, and the therapeutic efficacy will be higher. Bioavailability of the molecule will be enhanced when some molecules are having poor oral bioavailability because of the pre-systemic metabolism and self-administration possibility. These are all the advantages over the transdermal patches. Before we are going to discuss about the microneedle or transdermal patch, we should know about the structure of skin barrier also, how the skin is acting as a barrier to deliver the drug molecule. Actually, skin giving the highest advantage of preventing entering of any foreign molecule into the body. Actually, it is most important organ in our system, but it is a barrier for the transdermal drug delivery patches. The barrier is because of the presence of epidermis and dermis layer, and the actual barrier, several barrier nature is because of epidermis. Presence of stratum corneum, a horny heavy layer, and stratum lucidum, a translucent light translucent layer. Stratum granulosum, like granules are present, still the light can passage through it, and stratum spinosum, squamous cell layer, and then basal layer. These layers are going to give high amount of barrier to the to the movement of the drug molecules inside the skin. And apart from that, dermis pod is a complex system which composed of several organs like blood vessel organelles, like organs like blood vessels, glands, hair follicles, lymphatics, nerves, and fat cells are being present. And hypodermics still again going to give the barrier nature through subcutaneous nerves, vessels, lymphatics, sweat gland, and because of this, the delivery of drug will be typically problematic in case of transdermal delivery patches. Though the skin having barrier nature, the skin have certain ways to deliver the drug. Certain mechanism allows the drug molecule to penetrate through the skin and to reach the systemic circulation. For example, the drug may cross the cells. It can penetrate the cell and it can penetrate inside a cell, and then after transverse the cell, and the drug molecule will move towards the cell, inside the cell, that is intracellular route. It can be delivered, or it can use the intracellular space available between the cells. Rather than touching the cell, without touching or without entering inside the cell, it use the intracellular spaces, or the space between the cells to penetrate through it and to reach the systemic circulation. Or it can use transappendageal route through sweat gland or sebaceous gland. It can travel, the drug can travel and reach the systemic circulation to the subcutaneous layer. And through this route, it reaches these circulations. Before we are going to understand about the microneedle, how it is differ from the transdermal patch structurally, let's have a very small brief introduction. A transdermal patch typically have a drug reservoir, an yellow color reservoir like this. There will be a drug reservoir which delivers the drug, which supplies the drug for a prolonged period of time, and there may be some mixture with the permeation enhancer on the yellow mixture itself. And apart from that, there is a drug release layer. That means it may be a semi-permeable membrane which allows the drug molecule through it to reach the skin surface. There will be a semi-permeable membrane, and apart from that, adhesive layer would be there to adhere the patch onto the skin, and the backing layer also will be there to protect the reservoir from the external environment, and to continuously deliver the drug inside the human skin. Apart from that, a release liner, a plastic sheet or a wax sheet that can be removed before applying onto the skin. That release liner also being there in the transdermal patches. Though the structure is very simple, there are several kind of types also available. It's not a typical, it's not a only system available in the market. Apart from that, there are several other types. Like what we discussed is of second one, reservoir type system. We discussed there will be a reservoir and there will be a semi-permeable membrane, and there is an adhesive layer, and there is a protective layer, and a backing membrane. Apart from that, several types of transdermal drug system available in the market, like nitroglycerin patches are available directly on the drug molecules are directly dispersed with the adhesive layer, and there will be a backing membrane. That's it. No other semi-permeable membrane, no other extra system, extra parts are there in the patches. Apart from that, there will be a kind of matrix system, like rather than having a drug reservoir, there is a drug matrix in which the drug is distributed in a polymer matrix, and a backing layer with the adhesive layer and a release liner, like as shown in figure 3 here, as I'm indicating in the using the cursor. Matrix system also available. Apart from that, the drug, instead of dispersing the drug molecule in molecular form in a matrix system throughout the polymer matrix, drug may be dispersed in a micro reservoir, small reservoir, like a small globules or small crystals, small polymer beads might be entrapped inside, and that will give another advantage of higher prolonged duration of application, or to protect the drug inside the micro reservoir until being delivered. This many advantages may be available from the micro reservoir system. So basically, these four types of systems are available in the market with the transdermal drug delivery system. Apart from that, there is another thing that we need to discuss. To enhance the penetration of the drug, there may be possibility for adding some penetration enhancer. Starting from some solvents may be used like water, alcohol, or dimethyl sulfoxide kind of solvents, or some other solvents like glycerol or propylene glycol materials, or sometimes surfactant also used like anionic surfactant, non-ionic surfactant, bile salts, and other systems like urea or calcium thioglycolate kind of material were also being used as a penetration enhancers. And after that, let's have a very brief idea about how the transdermal drug systems are being manufactured in industry. When a transdermal drug delivery patches are being made, first a backing laminate is going to be entered inside the machine, and in the backing layer, drug or adhesive solution, whether it is of polymeric solution or it is of solution with adhesive layer, will be applied, and the roller here, when a roller is being placed, the thickness of the adhesive solution will be managed based on the requirement of the quantity of the drug formulation. After the roller is leveling its drug quantity, the solvent evaporation system is going to be applied over here. Whatever the solvent applied to dissolve the adhesive and drug, or adhesive, drug and polymer, or drug and polymer, that will be evaporated. And after evaporating over here, there will be an impermeable laminate, or maybe some kind of semi-permeable membrane applied over here, and it will be cut into small pieces, and it will be delivered. Let's have a very brief video before we are moving further to have an idea how the transdermal drug systems are actually being made in the industry. Let's have a very short video on it. Can the video is shared already? Can you see the video? Any one of you, can you please tell me that video is visible? Hello? Any one of you, can you please tell me whether the video is visible or not? Or let me share the video once again. Right. Actually, the drug molecules, the backing layer is entering from here, and inside the system, the drug molecule mixed with the polymeric solution or the adhesive layer is being entered, and the solvent evaporation is happening at this place, and with the roller and cutter machine will be applied over here, and small pieces of the transdermal patches are coming outside the system. Yeah, that's right. Please come later. I am in the meeting. And let me share the screen once again. Is the screen visible now? Can you see the screen?
Yes, sir, it's visible.
Yes, yes. Okay, fine. In such a way that transdermal patches are being made, and several other evaluations are required before we are going to apply this transdermal patches into the market. We have several evaluations. These evaluations I included on one slide, since the same kind of evaluations required for the microneedle patches also, starting from the thickness measured by using the vernier caliper, uniformity of weight by using high sensitive electronic balance, drug content determinations by using HPLC or LCMS kind of equipments, drug content or uniformity of test using the same kind of equipment, drug quantification, moisture content test or moisture uptake test by using the desiccators, or folding endurance by using folding endurance kind of equipments, tensile strength using a tensile strength analyzer, and tag property test, whether it is properly sticking onto the skin or not, by using the thumb tag test, rolling ball test, or quick ball test. In such a way, the tag properties, how it is adhering onto this skin, will be minimized. For example, when we are having a rolling ball tag test, the rolling ball is placed over on the slanting surface, and it will be released to roll on it, and there will be an adhesive layer over here, adhesive film layer over here. When it is rolling on the adhesive film of the transdermal patch, how long it is traveling. It is actually a metal ball. When it is rolling towards the slanting surface, it obtains some speed. After reaching the adhesive layer, it cannot move for a longer distance when the adhesive film or adhesive layer is very active. When it is having more adhesive property, the ball will stick on it. It could not able to move forward. So based on the distance traveled by the metal ball, the tag test will be measured. How efficient the adhesive film is, that can be measured. Instead of that, they can use a probe also. Like in the probe, the drug the patches will be adhered, as shown in this figure. The probe will be adhere on it. Whenever they are detaching the patch, how much force is required to detach a patch from the probe, that will be monitored. Or peeling tag test also applied. The transdermal patch will be applied on the metal stainless steel plate, and at 90-degree angle, the transdermal patches will be removed, and how much pressure is required to remove the patch will be measured. I'm sorry. And in vitro drug release, by using several methodologies like France diffusion cell or simple diffusion cells may be applied. Apart from that, there are some in vivo studies, starting from the irritation test, or any local reactions are going on. This kind of in vitro experimentation also required before we are going to apply the transdermal system onto the skin. In the apart from the simple transdermal regulatory system, recent advancement also came in the research level, but not majority of the patches having any recent advancements in the market. We can apply electrophoresis, electric charge. Minor quantity of electricity may be applied to increase the penetration of the drug molecule. Or magnetophoresis, we can apply magnetic force to increase the traveling of molecule inside the human body. Or iontophoresis may be applied. Ionic charge, a very small quantity of battery, by using batteries, some electric charge will be applied. Because of that, the ionic molecules are being allowed to move inside the human body. That is called as iontophoresis. Thermophoresis also applied, like by using the heat, we are disturbing the packing of skin layer, and the skin layer packing will be disturbed by using the thermal energy. Because of that, the drug penetration will be enhanced. Apart from that, ultrasound is also being applied to disturb the stratum corneum layer, or the arrangement of the cells on the surface of the skin, and the drug molecule will be penetrated. Apart from that, now a recent advancement, jet injector also is also very advanced system being used for the transdermal drug delivery system. And apart from that, let's start I think with this information, we can move ahead for microneedle patch. This much information is more than enough for a transdermal patch. Now let's have a discussion on the actual discussion of today's discussion, microneedle. Microneedle is not a new thing to the world, or to all of the faculty members and other participants joined here. They might have seen microneedles in their life, day-to-day life. They might have seen it. We have seen in the first image, it is given there is a tomato plant with its leaf or its stem, which have very small kind of projection, that is in micron size, which is of actually they are trichomes of the plant. Trichomes may be of uniserrate or multiserrate, or it is having some kind of glands with some different kind of structures are available. That is a pharmacognostical discussion on it. What are the structures of trichomes? What are the types of it? And here we have some small needles called microneedles. We can say that these are all micron size needles. Where we can exactly feel the microneedles when it is piercing our skin. When we are handling the sugar cane, when we want to eat a sugar cane with a leaf, and if you want to cut down the leaf from the sugar cane, sometime you may be accidentally pierced with a small size of needles. When it is piercing, you will never feel pain. Maybe afterwards, after 5 to 10 minutes, it will start itching. When you are start rubbing it, it will again penetrate inside the skin, and it will give a allergic reactions. It may be redness or itching may be produced. So that microneedles are not new to us. It's a very common thing which we have seen in our life. In absolutely, if you are saying it's a day-to-day life, we have seen microneedle, but we never thought of to use these needles to deliver the drug. There was a huge history behind in finding the microneedle based drug delivery system. But basically, I want to say from some points from my end that nature is always being the inspiration for many innovations. Whenever we are making any innovations or any inventions, when we make the nanoparticle, I used to see that the whole world is a kind of nanoparticulate system. Like it's not a nanoparticle, but it's kind of a globule kind of system. It's a spherical system, which is created by the nature. The whole world is like a small particle globe roaming around the whole system. Like that same way, we are preparing the nanoparticles also. In such a way, microneedles are also inspired from the nature. From the nature, by seeing that the trichomes of leaf, or by seeing the trichomes from the sugar cane, once a scientist might have inspired on it to prepare it. Here we can see exactly when we are comparing the trichome of a leaf and the prepared microneedle in our laboratory, there is no much more difference. If you are seeing the trichomes of a leaf, the structure may be varying, and the size may vary, and the shape may vary. It may be bent or it may be straight, something. But in case of microneedle, we can prepare, if you have prepared the microneedle in the laboratory, it would be of having specific size and specific dimension, and can be used for the delivery for the drugs through the skin. And with this information, we have several advantages with the microneedle, like no pain when we are applying the microneedle. The reason is, though it is having small needle, the size of the needle is varying from in microns, less than a millimeter. For example, typically in our laboratory, we are preparing 750 nanometer height of microneedles with a base breadth of 250 micrometer. With this 750 micrometer, the needle could not reach the sensing neurons or pain sensing neurons of the skin. That means the pain sensing neurons which are available at the dermis spot could not be could not be reached by the sharp needles. Because of that, we cannot feel any pain when we are applying the microneedle patch on the skin. So it's painless, non-invasive. There is no exercise of operations to install the microneedle patch onto the skin, and absolutely it improves the drug bioavailability in comparison to the topical application or to the transdermal patches. Since it pierces the skin completely, it pierces the top layer of the stratum corneum. It delivers the drug inside the dermis spot, and no pain when we are applying the patches like injections. Self-administration possibility. We don't require any medical personnel to apply it, and no medical waste. Simply the backing layer, which is of biodegradable polymer material, will be there, and that can be easily removed, and it can be destroyed. And no medical waste. Patient high compatibility of the patients, and ease of supplying the patches rather than the medications. If we have during the COVID time, if we have the transdermal patch with the COVID vaccine, that can be easily transported from one place to another place, and we do not require any additional medical personnel to apply on it, so that we can simply purchase a COVID vaccine patch from the medical shop and we can apply onto the skin, and our vaccination will be completed within 5 minutes without any help of medical personnel. Just we need to pay money, get the patch and apply onto the skin, and I'm vaccinated. That's it. So the whole India may be vaccinated in a day when these kind of systems are existing. Like when bulk manufacturing possibilities are there, at that condition, we can have the patches immediately from the medical shops, and we can purchase it and we can apply onto the skin, and the vaccination may also be completed, or any drug delivery application may also be completed. And we already seen that what is the advantage over the microneedle, like from the topical application or from the transdermal patches or from the hypodermic needle, it is having the advantage. For example, when we are applying the hypodermic needle, it penetrates through the stratum corneum, it penetrates through the epidermis and dermis, it delivers the drug in the dermis part. But at that point, when the drug is being delivered, here the red color thing, what you see in these are all pain sensing receptors, pain sensing neurons are present. Pain sensing neurons will be disturbed by the hypodermic needle, it piercing it, it disturbing the pain sensing receptor and then delivering the drug molecule. With this, the drug molecules are delivered with some pain. But in case of microneedle patch, this will not be there, because the microneedle patch cannot reach the dermis part. It will reach until it will cross the stratum corneum, it will cross the epidermis, but it cannot cross the dermis part. It will just deliver onto the dermis part or just above the dermis part to deliver its drug molecule, and it will not touch, it will never touch the pain sensing neuron, so that the pain will never be there while we are applying the microneedle patch. While we are seeing the history, actually the microneedle systems are being seen in the history before 100 years onwards. At a very early stage, the dental burrs, that means a small needles kind of structure, small projection kind of materials, they are called as dental burrs, are used to create some scars, create some pores on the skin to remove the scars, or to treat the hyperpigmentation, to apply the drug for hyperpigmentation. Though we are applying the drug for hyperpigmentation on the skin, it could not able to penetrate through the skin to treat the hyperpigmentation or scars. After surgery, when the scars need to be treated to make the skin into a normal structure, it can be used. It was used in 1900 year. 100th year, and then after that, microneedle literature is available between 1900, after 1900th year. The microneedle word is already reported in the literature, but not in the name of microneedle patch. In 1960s, drugs are being delivered through stratum corneum, and it was identified. Topical application was more intense in 1960s, and 1970 microneedle concept was introduced. Microneedle separate system were introduced into the literature, and research work were started. And in 1980s, first transdermal system was approved. The scopolamine drug was approved for the motion sickness in before 1980s. I think it is in 1979 or something. At the time, it was approved, and afterwards, the microneedle concept was demonstrated experimentally in nearly 1990s. If you see, within 25 years, the microneedle concept is being demonstrated, but not for the drug delivery purpose. It was used for correcting the wrinkles, or correcting the scars, or correcting the hyperpigmentation by using microneedle rollers. They reported, and the first microneedle manufacturing was started nearly in 2000 to get these kind of formulation, the microneedle roller kind of formulation into the market nearly in 2000. After that, dissolvable microneedles appeared into the literature, or microneedle array were used to pierce the holes on the skin for drug delivery purpose. For example, we may have seen that in the early literature, the vaccines are being applied using the microneedle arrays. When the doctors are trying to apply the vaccination, the microneedle small needles on the small plates were applied onto the skin, and it will be used to pierce the skin or to make a local wound on the skin, and on the local wound the vaccines were applied. Like Edward Jenner were also used the same kind of technique. He created the local wound on it, and then he applied the vaccine on the skin. But it was a macro wound. That means it's not like a micron size of wound. It was a macron size wound created by using millimeter kind of needle. But the micron size of needle were reported in the mid of 2000 to 2010. The first clinical trial started before 2010 for the clinical applications of microneedles for drug delivery purpose. Presently, it reached until the third level of clinical trial. Phase 3 clinical trials are going on for the delivery of vaccines. It is being applied now, but still it is not touching. It's not hit the market. We can soon expect that this kind of formulations will reach the market also. And now the manufacturing methodologies are being proposed, and commercialization of the microneedle patches are being attempted, and the commercial 3D printers are also reported to fabricate the microneedles to hit the market very soon. And these are all the types of microneedles. Microneedles might be of a solid microneedle, as I told you. It may be of simple metal needle to create a local wound on the skin. On the local wound, the drug may be applied, and the drug molecule can bypass the stratum corneum through the local wound or through the local pores created on the surface. The drug molecule may penetrate inside, and it can reach the systemic circulation. Or the drug molecule may coated on the microneedle. Instead of having the solid metal microneedle, it may be a polymeric microneedle. Some drug molecule may be coated on the surface of the polymeric microneedle, and it can pierce the skin to deliver the drug molecule inside the dermis part. Or the whole microneedle patches may be made up of dissolving kind of microneedle, and through the dissolving microneedle, the drug may be applied for the transdermal purpose. Or the transdermal patches made up of a hollow kind of microneedle. There is a reservoir inside the microneedle patch to deliver the drug or to fill the drug, and after filling the drug, the hollow microneedle patches might be pressed to inject the drug inside the human body, as shown in the end of figure A and end of figure B. The the hollow microneedle may also be applied. And when we are look at the preparation of the microneedle, it was a very simple preparation. As we seen that microneedle may be a new technology and an innovative technology for the transdermal purpose, but the preparation method is not that much tedious. If we have very few equipments, we can go ahead with the microneedle preparations. Like if you are having an acrylic sheet, the pores may be created, or the micron structures can be created by using CO2 laser or by using a laser beam. And we can use the mold. We can use the master mold to prepare the microneedle, or we can use lithography, like a chemical lithography or physical engraving kind of lithographic technique to use to prepare the microneedle master mold. Or 3D printer photolithography can be prepared, like layer by layer deposition or direct photolithographic technique may be used to create the master mold. We are preparing the master mold by using the 3D printer. Typically, we are having a very small setup for microneedle patch preparation, especially for preparing the master mold. Let me share that video also, or maybe you can see that to have more idea onto it. And once again, let me have the sharing first. Is the screen visible?
Yes, sir, it's
Uh,
visible, but it's in the slide mode. You need to
video
change
is
it.
not, yeah, video is not the same, right? I can see that.
It's okay, sir. It's okay.
No, no issue. Now I think you can see the video, right?
Yes, sir.
Yeah,
Yeah, here we have to prepare the actual microneedle in a 3D structure by using on-shape or some kind of CAD technique. And after preparing it, just we have to take the print by using the 3D printer. Here is the 3D printer. What we have, frozen mini 8K equipment right here we are having, and we can pour the resin onto it, and we can plug it whatever the structure we prepared. We can just like a word copy, like we can prepare, take the 3D structure, and we can just insert into the system, and we can select the print, and the printer will be processing for printing the microneedle. After printing, we require to wash it to remove the excess quantity of the resin, and the microneedle will be printed. And after printing, we have to wash it by using some washer kind of equipment, and here the washing protocol will be completed by using the system. Later, please. And afterwards, the cure also going to be done. Means after washing the monomer molecules, and it should be cured for with the UV light, so that it will be firm. It will not be flexible in nature. So the this will be placed onto a platform, and the UV light will be on, and that polymer, the photosensitive polymer, will cure it, and the microneedle patches will be made. The master mold will be prepared. With this, let me skip the presentation to the actual slide here. And after preparing the master mold, we have to place the master mold on a PDMS solution, and with that we can prepare a PDMS female mold. And onto the female mold, we can cast our polymer or drug solution or vaccine solution onto it. And above that, we can place the backing film layer also. And above the backing film layer, we have to after keeping the backing film, we have to dry it. And after drying, we can take the whole patch with the backing layer, as shown in the last figure. The dissolving microneedle will be taken out from the mold, and the tip dissolving microneedle may also be prepared. Instead of dissolving the whole patch, tips may be dissolved and the patch may be prepared. Or the microneedles may be coated. The instead of preparing the dissolvable polymer, if you are having PLA kind of polymer, water insoluble polymer, the microneedle will not dissolve. Instead, onto the microneedle, the drug may be deposited, and it can be applied for the transdermal applications. And hollow microneedles may also be prepared. We should have a bottom mold and a top mold, and after keeping bottom mold and top mold with a small space between it, and we have to inject the polymer and drug solution onto it, and the hollow structure of microneedle will be prepared. And with this hollow structure, we can fill the drug molecule, and then we can dry it. And after drying, or after filling it as such in the liquid form, or in the semi-solid form, we can use the hollow microneedles for injection. As we seen in the transdermal patch evaluation, in addition to that, swelling index need to be evaluated. How fast it can swell when we are applying onto the skin, and microneedle failure force or mechanical strength of the microneedle. Whether the microneedle is strength enough to pierce onto the skin, or it is bending, or it is going to break when we are applying onto the skin, need to be evaluated. And scanning electron microscopy may be additionally useful to see the micron structure or the surface characteristics of the microneedles. And film insertion onto the synthetic layers may also be evaluated. And in our laboratory, what we are actually working on, we prepared levothyroxine loaded PLGA nanoparticle, and then loading it with the microneedle for hypothyroidism treatment. And we prepared a clonidine loaded microneedle patches for hypertension treatment, and tetanus toxoid vaccine loaded microneedles were also prepared for the tetanus toxoid vaccination. And tretinoin is used for the melasma, a kind of hyperpigmentation disease. It can be applied. The microneedle containing tretinoin PLJ nanoparticles are being delivered for the treatment of melasma, and mometazone loaded microneedle formulations are used for the psoriatic treatment, and tolnaftate loaded microneedles are used for the fungal infections, and amylorphine loaded microneedles we prepared for the fungal diseases, and for dacarbazine loaded for melanoma, for skin cancer, and hepatitis B for vaccination. And we are also working on to get the COVID-19 vaccine to deliver through microneedle. Though the US-based research laboratories are trying to get the COVID vaccine to work on microneedle, still we are also trying to get the protein from antigen protein from the Pune to establish the microneedle preparations. And these are all the microneedle molds what we prepared, the 3D printed microneedle molds, and then female molds, and the microneedle by using a simple microphotograph, simple photograph by using microscope. And these are all the scanning electron micrographs of microneedles. And this is how we are preparing the female mold. Just we have to take the PDMS solution, and onto it the microneedles are being placed to prepare the female mold. On the female mold, we are pouring the drug. We are casting the drug and the polymer solution to create such kind of microneedle formulation. These are all the some other micrographs of scanning electron micrographs of the microneedle formulation. This is how we are checking whether our prepared microneedle can penetrate through the skin or not. Instead of taking skin, we are taking synthetic layers, synthetic polymer layers to check whether the microneedle can pierce the layer, and how many layer it can penetrating it. We are checking it. We can keep the stack of layer, and then we are applying the microneedle and checking until which layer it can penetrate it. Here, as we seen, it penetrated until 700 micrometer in the seventh end fold, and it could touch the eighth end fold, and eighth end fold is free. That means it can penetrate until 700 micrometer. And this is the images for dacarbazine loaded microneedle for treating the melanoma hyperpigmentation. And these are all the images of the coumarin 6 loaded microneedle which shows its structure. And this is the actual patch we prepared for dacarbazine loaded microneedle, which is evaluated for the skin cancer. That the evaluation process is also going on. And the microneedle mediated delivery for amlorolefin is also applied. This is used for the treatment of mycosis. It's actually an anti-fungal drug used to treat mycosis. And the amlorolefin loaded microneedles were also prepared, and the antifungal efficacy is being evaluated. And some microneedles for the psoriasis is also prepared. Mometazone furoate loaded microneedles were prepared. Actually, we prepared the PLJ nanoparticle which contains the mometazone, and then the nanoparticles are being loaded onto it. This is the image of the optical image of the mometazone loaded PLJ nanoparticles and loaded onto the dissolvable microneedle. And these are all the photo scanning electron micrographs of the momentozone nanoparticle loaded microneedle formulation. Apart from that, there are several other data present we have like the tetanus toxoid and hepatitis loaded and other small molecule drug loaded microneedle also prepared, and the data are not included in it. Even these publications also not this data also not published into the in the journal. That's why I'm having a very limited number of slides from our own data, and more number of slides were given on the theoretical part, and then whatever the protocol that we are following. With that, I can say microneedles or a very easy carrier system which can deliver the drug through the skin, and hope we can get the microneedle patches very soon in the markets after completion of the phase 3 clinical trial. We hope within a short period of time we can have the microneedle patches in the market, and it is a wonderful tool for delivering the vaccines which require very small quantity to be delivered through the skin. And apart from that, if the drug molecules require to be delivered for a prolonged period of time, like a transdermal patches, this microneedle patches are also useful and much better than the transdermal patches. For example, transdermal patches can deliver the drug through diffusion only. Instead of the transdermal patch, if you are using microneedle loaded transdermal patch, microneedle patches, it will deliver the drug molecule across the skin much efficiently than the simple transdermal patches in the market. With that concluding remarks, I'm ending with my presentation, and I'm happy to answer your question. And thank you each and every one of you to spend your valuable time to listen my presentation. If any of you require my help from my laboratory to prepare the microneedle or to start your research work on the microneedle, you are always welcome, and I'm happy to assist you without any expectations. With this, I conclude my presentation. And thank you, Dr. Yunesh, for offering me an wonderful opportunity to share some of my findings with the audience. Mostly, I think they are from faculty audience. Though you are having a very advanced research laboratory, this may add one more knowledge to your lab. And I welcome all research scholars and then faculty members to continue the research on microneedle as well. Thank you so much for offering me the wonderful opportunity. I hand over the session to Dr. Somnath Roy sir or Dr. Yunesh to conclude the session. Thank you. Thank you for offering the opportunity.
Okay. Thank you, sir. It was really a wonderful and enriching session, so to say. And this microneedle concept, personally, I liked it, very enlightening, and I didn't have much knowledge, and it was very fruitful for me. Thank you once again from on the behalf of organizing committee. So the house is now open for questions. So I request the participants to come up with your questions. You may directly read out, or you may wish to put it in the chat box.
Yes, sir. We have provided the permission to unmute.
Good,
Yes.
good morning, sir.
Yeah, ma'am. Good afternoon.
Good afternoon, sir. Dr. Vijay Kumar, sir. It was like an enlightening session. And I have one basic question, like these micro needles are available in the hospital, sir, to treat patients currently?
No, ma'am. No. Actually, microneedle formulations were not yet approved by any regulating body, though it is in India or in anywhere in the world. No such formulations were approved as of now.
Okay, sir. Okay, sir.
Yes,
And another one question, sir. Can you take students for internship or project in your laboratory?
We are already having, ma'am. I think Dr. Gaurav from the Lotte University, he already sent students to the laboratory. I'm always welcome. I always welcome the scholars or internships.
Okay, sir. Thank you, sir. Thank you so
You
much.
just write an email, ma'am. We will be happy to have students.
Yes, sir. Sure, sir. Sure, sir. Definitely, sir. Thank you.
Sir, hello. Am I audible?
Sir, good afternoon, sir.
Are audible.
Yeah, actually, sir.
Hello. Good afternoon, sir.
Ji, ma'am. Please go ahead.
Sir, for which type of drugs these microneedles are portable, sir? Means which class of drugs or which type of drugs are suitable for making them as microneedle type of dosage form?
Exactly, ma'am. Actually, like as I told, that vaccines are the very best candidate for the microneedle, which actually we could not able to deliver in higher quantity, like 10 milligram, 20 milligram, 50 milligram, 500 milligram cannot be delivered through it. But if it is in micron level, like antigens are required 10 micrograms or 20 micrograms of antigens are more effective. So we can go for the drug molecules or vaccines having lesser quantity of dose. Maximum, as per my calculations, we can go for loading 5 milligram of the drug with these small microneedle patches. So the dose should be very small, and the application may be extended for prolonged period of the time. Also can be managed.
Means can we go for anti-cancer drugs, sir, for making them as microneedles?
Absolutely, we can go. We are also working on dacarbazine for treating the skin cancer. So the drug molecule, anti-cancer drug molecule, having very small dose can be used for the to deliver it through microneedles. It may be used.
Hello, sir. Am I
Ji, ji.
audible, sir?
Yes, sir, you
Actually,
are
sir,
audible.
I am Dr. Sadada Prasanna Malik's PhD student. So I am using his account only for asking you a question, basic question, sir. Sir, can we use microneedles for, in deep injury and trauma injury, these things like there are bioscaffolds and biomaterials are used in those
कभी मुझे
cases?
फील होता ही था कि मतलब है भी है मतलब
Using microneedle for
Sir, trauma injury, deep injuries, wound
uh,
healing, these things.
wound healing property for deep injuries. Actually, microneedles cannot be used directly to deliver the drug onto the deep injury because
Okay.
microneedle can deliver locally up to 1 millimeter, and systemically we can deliver it. If we want to disperse the drug throughout the body, we can use it, and if we want to deliver the drug at the local area, we can use it. For wound healing, it can be used, but very limited application will be there. But in case of scars, after wound is healed, if you want to heal the scars, it can be used. And for wound healing, you can deliver the drug. If any transdermal kind of applications, if you want to make, definitely it will be useful. But it cannot deliver the drug to a larger quantity, like antibiotic kind of molecules cannot be delivered in larger quantity in the wound healing. And
Okay.
limit the problem is, we could not use the microneedle for delivering more than 10 milligram of drug quantity.
Okay. And sir, one more question is then like melanoma, you presented that in melanoma you are using these patches and microneedles and these things. So in breast cancer also, sir, there is a certain, I mean, the lesion is visible from the skin and these things. So can we use this kind of thing in breast cancer
Definitely.
also?
Definitely, you can use. Actually, we need to understand the basic that the
Yeah.
microneedles are not for the local application.
Yeah.
Whether the the tumors are visual from the top,
Mm.
or it is not visual from the top.
Mm.
Even if the tumors are located interior part of the
Yeah.
body, the microneedles can deliver the drug molecule throughout the body
Yeah,
systemically.
because it is just a drug delivery system only. Yeah.
Exactly. Just
Yeah.
like a transdermal patch delivery, we
Yeah.
can use it. And if we want a local delivery of molecule to a larger extent, still
Yeah.
microneedles are more helpful.
Yeah. Thank you so much, sir. Yeah.
Welcome. Welcome.
Sir, which type of polymers are used as microneedles?
It depends on the type of system, ma'am. If we require insoluble microneedle, coated microneedle, we require insoluble polymer. We can use PLA, polylactic acid polymer, or PLGA, or PG, sorry, not PGA, polycaprolactone kind of polymer can be used. If we want a dissolvable microneedle polymer, the drug and the polymer matrix should dissolve after application on the skin. We require the soluble polymer, like we can use polyvinyl alcohol, or we can use polyvinyl pyrrolidone, or we can go for hyaluronic acid, which is more compatible with the skin. This kind of polymer may be used for the soluble, dissolvable microneedles. And most importantly, the polymer should be biocompatible, and it should not have any local irritation, and it should not have any local reactions onto the skin. That is the only limitation. We can use the polymers.
Which polymers will have that ideal property? Which polymers will have that ideal property?
Yeah, most widely they are using PVA, PVP, polyvinyl alcohol, polyvinyl pyrrolidone, and hyaluronic acid for dissolvable polymer, as I said. And for the insoluble polymer or coated polymer, they are using poly lactic acid, PLA, and PLGA were reported, ma'am, widely.
Thank you, sir. Thank you very much, sir. Very informative session, sir.
Welcome, ma'am. Welcome.
I hope we can go ahead.
Thank you, Dr. Vijay Kumar, for this wonderful session. So please join me to thank Dr. Vijay Kumar virtually. I just put a clap sign as a gesture of appreciation. Okay, so now I request Dr. Yuvanesh sir to kindly hand over the memento to sir. I just share the screen. Uh,
Vijay sir, just kindly.
yeah, I have managed it. Yeah.
Thank you. Thank you so much, sir. Thank you so much, Somnath sir and Dr. Ivnesh, for sharing such.
Sir, please accept the token of our gratitude
Yes,
from
thank
our.
you. Thank you, Dr. Ivnesh. Thank you. Thank you so much. And thank you so much for providing a wonderful opportunity to at least meet the respected faculty members and most renowned researcher also in the wonderful platform. Thank you so much.
Thank you, sir.
So, so now good to go. So we move further with our next speaker of the day. So please join me again virtually to welcome our next speaker, Dr. Sarita ma'am. I think she's already on board. So before we start the session, let me give a brief introduction of Dr. Sarita. Dr. Sarita, she did her master's MTech in biomedical engineering from Motilal Nehru National Institute of Technology, Allahabad, Prayagraj, Uttar Pradesh. Then she joined Guru Jambeshwar University of Science and Technology, Hisar, Haryana, as a teaching associate. Now she's working as an assistant professor at biomedical engineering department at Deenbandhu Choturam University of Science and Technology, Mudthal, Sonipat, Haryana since 2013. She did her PhD in biomedical engineering from MNNIT, as I already mentioned. Her research interests encompass a wide field including biomedical engineering, biomaterials, biomechanics, and nanomaterials. She has already published multiple research papers in reputed peer-reviewed journals. So I again welcome Dr. Sarita ma'am on board. Uh, Hello, ma'am. So the stage is yours. Ma'am, you need to unmute yourself.
Am I audible?
Yeah, you are audible.
Just 1 second. Good afternoon, everyone. Myself, Dr. Sarita, Assistant Professor in the Department of Biomedical Engineering at Deenbandhu Choturam University of Science and Technology. And topic of today's talk is different approaches in bone tissue engineering, where we explore the different advantage and disadvantage of various methods which we use in bone tissue repair and regeneration. First of all we know there are lots of cases of bone fractures every year if we talk about the previous data or recent data. And a bone fracture is the simple discontinuity in the bone. And if we talk about the healing of a bone, it includes various factors that affect the bone healing in a particular patient. That is normal nutritional deficiency, aging process, and infection at the bone fracture site. And in that case where we are unable to recover the fracture, we need bone grafts, and where we use various type of bone grafts in case of complex fractures, disease. And traditional surgery has been done for the healing the fracture or to uh, process the fracture healing. Various methods have been used, and where there are various type of grafts also used for the bone healing, like autografts, allografts, xenografts. And in this case, autografts, where we use the patient own bone for the healing of the fracture or to transplant the own bone of the patient from the low load bearing site. It have the advantage of osteogenic, provide the osteoblast and osteocyte of the own patient. In that case, we have the disadvantage of two site surgery, donor site morbidity, And in second case, we have the allografts. In allografts, where we use the same biologically compatible person bone for the grafting. It have the advantage of the second surgery and readily available for the patient. And disadvantage is the immune rejection. As we all know, biocompatibility is the main criteria for any type of transplantation. Next is the xenograft. In xenograft, we use the interspecies grafting for the healing of the bone fractures. But it have the disadvantage of the rejection, ethical issues, and cross species contamination. And in last, we have the synthetic substitute for the bone healing. For example, we use the different type of synthetic substitute made up of the material like metal, ceramics, and all. They have the advantage of the bone strength and integrated with the normally available bone, but it have the disadvantage of the poor adhesion and known biodegradability of a particular synthetic substitute. There, here we have the different type of challenges like non-union fractures. 5 to 10% of the fractures are failed to heal, which lead to the disability to the patient. Then we have the autograft complications like gold standard, yet autograft have the 20 to 30% complication rates and limited supply. And allograft have the risk of the immuno rejection, as we know. So in that case, we have the essential parameters which we have to study before bone grafting. First one is the osteoinduction, osteoconduction, osteointegration, and osteogenesis. It must be easily accepted by the body. Here we have the multiple approaches to consider the to heal the bone fractures. Initially, as we all know, we are familiar with the first approach, that is the surgical approach. In surgical approach, which is easy to work and economic, and but in this case we have the problem of histocompatibility issues, risk of the infections, and donor is not available easily for the allografting, and donor side morbidity in the autografts. Then we have the cellular approach. In that case, we use the cell coated implant for the bone healing. It have the risk of the cell flush out and have the issue of pore size and porosity. And last, we have the biomaterial and tissue engineering approach, which is ongoing recently, and lots of research is going on the biomaterial and tissue engineering approach. It have the good biocompatibility and good cell adhesion at the site, required site. It have the risk of immunogenicity. As we know, all have the any type of demerits and complications like complex procedure. This is the demerits of different type of bone grafting procedures. As we know, in case of autograft, we use the patient own bone. In case of allograft, we use the bone from the one person to the second one person, and xenograft we use the interspecies. And here we use the different type of surgical approach for uh, healing the fracture site. Here, first one is the surgical. Then second case, in this case we use the only mesenchyma stem cells, and stem cells have the ability to recover the fracture site. And in biomaterial approach, we have scaffold. We have different type of hydrogels that have been used for healing the fracture bone. And in first surgical approach, we have lots of globally, we have lots of surgery have been done, which give the uh, major turnover to the medical industry by using these surgical approach. But it have lots of limitations like multiple surgery, high infection risk. And in cellular approach, as I have uh, discussed, here we use the mesenchyma stem cells because these type of cell can generate any type of cells. For example, it can convert to the bone tissue cells, and it have the challenges same, limited limited stem cell source and all. Second one is a second approach, same cell source advantage clinical trials. It just moved to the phase 2 and 3 trial for the fracture of known union site. And here is some data which is comprises the different type of biomaterials have been used for the bone fracture sites. For example, here we study, here we can see that red barcode is showing the bioactive materials, then we have the smart materials, then we have the hydrogels, different type of hydrogels, then engineered tissues and organs for using for the bone tissue engineering. And this is the history of the biomaterials from replacement to the regeneration. Initially we see that where we use linen material thread for the wound healing and sutures, golden wire, golden plates, and which is evolving from different using different ceramics and polymers. Then we have various type of biomimetic materials, hydrogel, 3D printed engineered tissues and organs for the study. And next we use different type of growth factors and signaling for healing the bone. For example, we use BMP2 and BMP7 essential bio growth factors for the bone healing. And in this case we use these type of hormones, these type of growth factors using various type of vectors, and we just impose these type of growth factor in the scaffold and just implant the scaffold at the required site. But we face various type of challenges, like it have the short half life which move to the side effect to the body. Then we have bioreactor based approaches. In bioreactor based approaches we just regenerate these type of bone tissues in the bioreactor, where we use the perfusion based bioreactor. And in that perfusion based bioreactor, when we use these type of bone tissues, it will automatically generate, and then we apply some type of growth factors in case of signals like various type of growth factor like BMP7, BMP2, and it have the application for large construction. And here are some fabrication methods also for scaffolding, like electrospinning, 3D printing, additive manufacturing. And in the stem cell and gene therapy integration, we first of all we use the MSCs, as I have discussed previously. Next, we have the induced pluripotent stem cells, that iPSCs, which offer the unlimited cell supply. Here we use different type of cells which can differentiate easily to the required cell types. Then we have the growth factory delivery, like uh, discussed earlier, BMP2 and various type of growth factor. And last one is the gene editing. In gene editing, we just edit the gene of a particular bone cell DNA, and we just apply the different type of growth genes there, which is responsible for the faster growth of the cell, of faster growth of the bone cells. And as we know, all tissue engineering multidisciplinary area, which include the concept of engineering and biosciences for the therapeutic surgery. And in this case we have bone tissue engineering, where we use various type of cell carrier biofactors for the cell regeneration. It include the four core principles, like it include the cells, scaffold, functional bones, and signaling molecules. And in biomaterial approach, we have the first one is the scaffold. Um, Scaffold is the extracellular matrix which includes the cell attachment and growth. It must have the pore size 100 and 100 micrometer. Ideal scaffold must have the biocompatibility, biodegradable, surface chemistry appropriate. And here we use the scaffold at the bone fracture site, and diagram showing the healthy bone uh, regeneration. These are the various material have been used for the uh, bone materials. Excuse me. In this case, we have the different polymers, metal, ceramic, and composites. This is a scaffold based strategy, and the scaffold made up of different materials like polymer, ceramic, and composite. And if we talk about the hydrogel synthesis, we have synthesized various type of hydrogels which have been used for the bone tissue engineering. First one is the chitosan based hydrogel. Second one is the hydroxyapatite chitosan based hydrogel. And third one is the high hyaluronic chitosan based hydrogel. And last one is the hydroxyapatite high hyaluronic and chitosan based hydrogel. And this is the hydrogel synthesis. And in this case we just use the chitosan and dissolve the chitosan overnight by using magnetic stirring. Then we add hydroxyapatite, hyaluronic acid based on the different type of hydrogel composition key, what type of hydrogel we are synthesizing. And then we use the homogenizer for synthesizing the hydrogel. Here we have the different type of hydrogels we have designed in our laboratory. And this is the various type of synthesized hydrogel which we have used for the bone tissue engineering application. And all hydrogels have the thermal stability, which is a mandatory property if we use these type of hydrogel in body implantation. And in four cases, hydroxyapatite, hyaluronic, and chitosan based hydrogel have more rough and large surface area than the other type of hydrogels, which help the bone cell to adhere and grow faster at the surface of the hydrogel. And different hydrogel have the pore size which support the cell seeding, tissue growth, and nutrition supply with waste disposable. But in this case, HAP, HA, and CS hydrogel, it's showing the highest porosity in comparison to the other type of hydrogels. And all hydrogels have the sufficient percolation capacity, because if we add something like growth factor drug bone cells to any type of hydrogel, it must be percolate at the core of the hydrogel. That is why we have done study of the percolation. And this HAP, HA, and CS and chitosan based hydrogel have the high percolation rate in comparison to other two hydrogels. And if we integrate these all approaches with latest trend that is going on. First one is the 3D printing. In 3D printing, we designed patient specific scaffold, which is we use which use different type of cells, growth factors, and biomaterials. And these designed bone grafts have been implanted at the required site. Second one is the gene editing. Gene editing in this case, as I have discussed earlier, we just required a specified DNA and we replace the mutated place. We just replace that with the required editing, like for bone growth, a faster healing. These type of gene can be replaced with the another one. And last, we use different type of co-culturing and done with the different type of control growth factor delivery at the site, so that there is a lesser amount of nutrition need and larger construction of the various type of scaffold which we have used for the further applications. And here we different type of growth factors, role of the growth factors, and clinical examples like BMP2 and delivery at that specific site. But there is also some uh, limitations, like it need FDA approval and trials, different trials, human trials, animal trials, personalized medicine. If we uh, give a solution to a particular patient, it reduce the time of the surgery. Challenges, cost effectiveness, and long term efficacy data for the broader adoptions. And these are the various challenges. And future direction is smart scaffolds, active drug delivery, gene therapy, AI driven designs, in situ tissue engineering, and integrated approach. As we discussed, integrated approach include the tissue engineering, material science, genetic engineering for the further advancement in that area. Advanced scaffold, improved scaffold design, angiogenic factor delivery, 3D printing. And here we have the references. Thank you. Any question?
Okay. Thank you, Dr. Sarita ma'am, for your wonderful presentation. So I open the house for discussion. So participants are requested to come up with your questions, or you may prefer to put it in the chat box as well. I see people are posting with a lot of appreciation messages. I see Amit Kumar mentions it as a nice session. I don't see any questions so far in the chat box. So participants are encouraged to ask your questions to ma'am. Okay, if there are no more questions, then again, please join me in putting some appreciation reaction for Dr. Sarita's wonderful talk. Thank you, ma'am. Thank you for sharing your work. And I now request our HOD, Dr. Gaurav Kumar sir, to kindly present the e-memento to Dr. Sarita ma'am.
Thank you
Thank
so
you
much,
so much.
sir. Thank you,
Thank
Gaurav
you.
sir.
Thank you, ma'am, for accepting our invitation. Thank you, Somna sir, for inviting me. Ma'am, kindly accept this e-memento as a gratitude from the organizing committee member and Galgoti University. Thank you.
Thank you so much, sir. It's so nice. Thank you.
Thank you.
Okay. So thank you once again for the wonderful presentation. And now we move on further with our fourth speaker, Dr. Tayyab Khan on board. Welcome, sir. Welcome on board. So before we move further with his presentation, I take the opportunity to introduce Dr. Tayyab Khan in front of the audience. So Dr. Tayyab Khan is currently working as an assistant professor at the Indian Institute of Information Technology, Sonpat, an institution of national importance under the Ministry of Education, Government of India. He received his engineering degree in computer science and engineering from Gautam Budh Technical University, which happens to be our close neighbor of Galgotia's University, Uttar Pradesh, India. He then completed his MTech and PhD in computer science and technology from the School of Computer and System Sciences in Jawaharlal Nehru University, JNU, New Delhi. Dr. Khan's primary research interests encompass wireless sensor networks, IoT, network security, machine learning, and multicast communication. His extensive academic background and research expertise have led to the publications of numerous research papers in highly reputed SCI SCIE index journals. Recognized for his scholarly contributions, Dr. Khan has an remarkable H index of 12 and an I-10 index of 13. He is actively associated with multiple prestigious journals from IEEE, Elsevier, Wiley, Springer, etc. as a reviewer, and he provides critical evaluations that uphold high standards in scholarly publications. His role as a reviewer demonstrates his commitment to the advancement of knowledge in this field. Dr. Khan has organized several international conferences like NetCrypt series, faculty development programs at national levels, showcasing his leadership and dedication to academic excellence. Dr. Khan has delivered keynote addresses at numerous faculty development programs and has also served as session chair at several prestigious international conferences such as AutoCom 2024 and ICI-CCT 2024. His current research focuses on intrusion detection, information security, data science with machine learning, and IoT, information technology, areas that are pivotal in today rapidly evolving technological landscape. Throughout his career, Dr. Khan has been honored with several awards highlighting his impact in science and engineering. Notably, he received the Young Scholar Award at NetCrypt 2019 and the Young Scientist Award at NetCrypt 2021. He was also endowed with Best Teacher Award 2022, acknowledging his outstanding research achievements and reinforcing his reputation as a leading researcher in the domain. So I welcome Dr. Tayyab Khan to this faculty development program. So, sir, welcome to the session, and the stage is yours.
Thank you, Dr. Roy. Thank you so much for my kind introduction. My screen is visible to all faculties.
Right now, no, sir. It's not.
I'm going to
I mean,
share.
I think you need to put it in the full screen mode.
Okay, entire screen. I am selecting entire screen shares.
It's in the slide mode right now.
Sir, I am sharing. I am clicking on
Put it in
sharing
the,
button.
Sir, actually your screen is visible. Kindly put
uh,
it in full screen mode.
Okay, full screen.
It is like
Screen mode. Yes, sir. Now it is showing.
Sir, your screen is visible. Kindly put it in full screen mode.
so you just scroll your cursor in the bottom right where you find it
Okay, sir.
I mean, it's in the slide mode now. It's not in the full screen mode. You just need to click on the, yes, just a bit left. You can put your, yeah,
Next to
just
that,
next
sir.
to it,
Next
the
to
next
that.
one.
Next one.
Yes, yes.
Okay, sir.
Perfect.
Fine.
Okay, sir.
Go ahead.
Now it is visible, sir.
Yes, yes, sir. Please go ahead.
Okay. Let me start with trust management in sensor-enabled IoT networks. First of all, let me discuss with some outline regarding our topic. First, we will discuss what is IoT. Then we will discuss motivations and limitations. Then we are going to discuss what is trust and reputation in sensor-enabled networks. Then we will discuss some problems and what is our objective to achieve today. And then we will discuss some trust evaluation methods and how to design trust models. Then we will conclude what is the significance of trust models in IoT-enabled networks. So first of all, IoT is a system of connected objects like sensor, devices, machines, or appliances that are connected to the internet that can collect, sense, and exchange data without human intervention. So we can say IoT means everyday thinking, talking to each other online to make life easier, smarter, and more efficient. There are generally four layers in IoT: sensing layer, network layer, data processing layer, and applications layer. Sensing layer sends the data from the environment and it passes to the network layer. On the network layer, there are some routers or gateways that forward the information to data processing layer, where there are some CPUs, machines, and then data is processed and forwarded to the application layer for further uh, information. These are some characteristics of IoT, like they have limited compute capabilities, low cost, small size, power consumption is low. They have some responsiveness, reliability, heterogeneity, scalability, and mobility. These are some characteristics of IoT devices. If we see the applications, IoT have applications in many areas. Like we have its application in industrial IoT, medical, where sensors are on the human body. They sense the temperature and the other symptoms of a human. And home automation, you can control your home appliance from the home, mobile using IoT sensors. You can use these in defense and business also. WSN, IoT, and M2M are closely related to each other, but I think IoT is a broad domain out of M2M. M2M is machine-to-machine communication. So this is a small communication between two devices. WSN means we are connecting or communicating the same type of sensors, but in IoT, number of sensors may be heterogeneous. Means different kind of sensors, they have different capabilities, different power, different configurations. They are communicating and exchanging data, and data is going to store on cloud. But in WSN, generally we use some internet, and data is going to end user. There are some research challenges in IoT, like we have limited memory storage, we have security and privacy concerns, we have limited power, unreliability of communication, we have limited bandwidth, we have some routing issues, transmission errors, cost and size, quality of service. These are the research challenges. So out of these challenges, we have some limitations like security. IoT networks are not fully secure as compared to wired networks. Hackers can easily hack the networks. There are some propagation delay that is almost 5 times more as compared to territorial. Some battery issue and the speed is because this is a wireless network, so there are some speed and connectivity issue. The range is very short, 5.5 m to 70 meter, and other like these are the limitations of IoT. These are some sensors used in uh, environment, motion, communications, energy power sensors, and industrial IoT sensors like DHT11. These detect abnormal environmental data and inconsistent across nodes. If you are deploying a sensor in a motion position sensors, so you can use ADXL345 accelerator sensors. It is used for monitoring mobility patterns and detect misbehavior or node compromise. These are the sensors we can use in different domains. Now, our the main focus is on what is trust and reputation systems. So, what is the trust, types of trust, and trust properties? We are going to discuss these topics in today talk. If we discuss about the trust is a subjective relation among the entities, while reputation is an opinion about an entity. Trust specifies the reliability or the trustworthiness of an entity. It ensures that an entity is secure and reliable. So we have categorized trust in different categories like based on observation, based on property, based on cooperation, and based on the place of usage. Direct trust means we are communicating with you, or a device is communicating directly with another device. That is direct trust. Indirect trust means we are getting feedback from other device or other person. Like social trust, quality of service, whether the data is transmitted, the quality of the service is good or not. Communication trust, like whether the devices are communicating or not. And like they are interacting or not, this is communication trust. And data trust means that the data is transmitting, that is completely transmitted, or some packets are dropped. So these are the categories of trust. We can use any one of the category in our model. These are the some basic terminologies related to trust, like what is trust value. Trust value is a score, or it's a level of quantification of beliefs of one entity towards another entity. It may be between 0 and 1. Like if a trust value is 0.5, we can say that it is 50% trusted person or trusted devices. Trust evaluation means we are monitoring the behavior, estimate the trust value, and then quantify it into highly trusted or trusted or distrusted in 3 categories, or you can qualify in more categories. Trust model are the methods to estimate the trust score. And trust management is seen generally, they provide the update trust, because trust has to be updated periodically. So trust management provide the mechanism or algorithms that can be trust can be updated dynamically. So these are the direct trust I have already discussed. These are the basic terms we will use in next slides. So the importance of trust evaluation in sensor-enabled network is it provides the secure communications because it detects the faulty nodes, reliable data sharing because it ensures only trustworthy data is collected and forwarded. It improves routing, reduce energy loss, and avoid weak nodes. It allows the nodes to evaluate trust locally without central control, and it evolves with interaction, helping the networks to adjust the change. So this is a topic that is can be used in almost all the engineering applications, because trust has to be calculated very carefully, and we generally take decisions based on the trust value. Either it is a person, it is a device, it is a machine, it may be a car, whether it will go or not. So we have to calculate trust of everything. Communication trust generally we say that whether the sensor nodes are communicating or not. So indirectly trust if sensor node X is communicating with sensor node Y, and sensor node Y is forwarding data to another node X, then we say that it is successful data transmission. Indirect trust means sensor node A is communicating with sensor node B, and it is taking feedback from sensor node C from about sensor node B. So this is an indirect trust. Data trust, whether the data trust measures the trust of the data created and manipulated by sensor nodes, which can access the fault tolerance and consistency of the data. Similarly, we have to calculate the energy trust, whether a node has sufficient energy so it can monitor the environment, it can capture the information from the environment, so it must have the sufficient energy to complete the task. These are the properties of trust. Trust is reflexive because every entity, either is a human or is a machine, it trusts itself. Asymmetric means if a device or a person trusts on another person, it is not guaranteed that another person is also trusted to a person A. Partial transitive, if a person trusts on person B and person trusts on person C, then it is not guaranteed that person A will be trust on person C. So it is partially transitive. And trust generally calculated in terms of context. And the last one is property of subjective. So these are the two terms like subjective and objective. So we will discuss subjective means trust is generally subjective because it is based on the opinion, not on the fixed rules. If anything is based on the facts, measurable evidence, and unbiased observation, rather than the personal feelings, opinions, and interpretation, that things refers to the objective. If it is not on the based of the facts, then it is considered as subjective. So trust is mostly subjective because it depend on the personal judgment, past experience, and individual perceptions. So this is a complete picture about the trust in Internet of Things, like what is the motivation, what is the design criteria, what are the different types of trust we can use, what are the attacks against the trust models. So motivation is like first model is the most important motivation is security and privacy. Then reliability, data sharing, heterogeneity of the devices, and design criteria, we have to use accuracy, latency, security. We have to use lightweight trust evaluation methods. It must be energy efficient, fault tolerance. We can use direct trust, indirect trust, behavioral trust, data trust, social trust, and there are many other type of trust that depend on the in which context we are using. And these are some attacks on the trust model, like bad mouthing attack, on-off attacks. In on-off attack, a node changes its behavior after a periodic amount of time. Collision attack means some nodes are together attacking on a particular node. So these are the attacks that exist in IoT networks. Let us discuss how to calculate the trust. First, we calculate the direct trust. Direct trust means first-hand information. Then we collect the second-hand information, and we can collect other information also like energy trust. Then we combine these trust and we apply the weights. We can give equal weights, or we can use some algorithms to dynamically provide the weights, and then we calculate the trust. Based on the trust value, we calculate we take the decision whether the node is trustworthy or not. These are the some methods used, like these are the broadly classified into three methods, like analytical, bio-inspired, and socio-inspired. So we can use weighting method, rating, or we can use fuzzy logic, graph theory based. So each approach has its limitation and advantages. So we have to analyze these methods in detail if we want to use any methods in future. So let us discuss first the elements of the trust model. First model is the information gathering. Then we have to score it. Then we have to use what entity we are going to select. Then we have to perform transaction between the sensors, and then we have to, if nodes are communicating in good manner, then we have to reward. As in our lives, we do communication with the students or with the some faculty members or with some friends. If we are interacting frequently and there is no misbehavior, then we reward. Otherwise, we give some punish. So in the same similar manner, trust models works. Now, what the main important what is the reason behind the erroneous sensor data? why the problem is data, from where this problem is coming. So problem can be coming from unintentional errors, like hardware may be broke, sensor is incorrectly attached, or CPU crash, because these are the unintentional error that can be insufficient memory. So trust value can be reduced due to this unintentional error. But there are some intentional errors, like attackers attack on the data. They drop some packets from the data, or which they change the information, corrupt the data, or they hijack the data, destroy the data. If the data is destroyed, or due to insufficient memory, proper data is not stored in the memory, so how to take proper decisions? So we have to check the errors before this trust evaluation methods. So these are the reasons for corruption of erroneous of sensor data, unintentional and intentional. So we have discussed what the problems in the previous slide. So these are the some open-ended questions in your mind, sir. Which deployment scheme may be best? How to improve cooperation among sensor nodes of IoT? How to protect a sensor network from severe internal attacks? How to detect malicious sensor nodes? How to punish and rewards? How to improve the lifespan of IoT? How to find reliable nodes, or how to find shortest path? These are the questions that may be arise in your mind. So one by one, we will discuss how to design a trust model. So our first objective is design a trust model. Then we will provide a trust updated algorithm, and then we will apply into a routing algorithm. So these are the three objectives for this session. First, we will design a trust model. Then we will provide a trust management algorithm for updating the trust value, and then we will provide the routing mechanism. And then we will discuss in the last what is the importance and significant impact on the world or on the social life. So we are going to design a light weight trust estimation scheme for large scale IoT that employs clustering. We have divided the nodes like in a hierarchy in any organization. One is Bosch, and there are other clusters like other branch, computer science branch, EC branch. In the similar manner, we have divided the IoT networks into clusters, and within each cluster head will calculate the trust value of nodes. Then from all the clusters, they will communicate to directly the boss. That is the uh, that is uh, base station or gateway node. That will calculate the trust value of all the cluster heads. But before computing this trust, we have some assumptions and limitations like clustering. So we are using a clustering using the leech algorithm. It is a very popular algorithm for clustering. We are assuming that our channel is employing a key management scheme to secure from the external attacks. We have to assume domain of the trust value, that our domain of the trust value is 0 to 1 or 0 to 10. If we take 0 to 100, then to store the 100, we need some more number of bits. That's why we have taken some lower range. We are using a timing window mechanism to observe the records whether the interaction is successful or cooperative or not. And we are going to use a base station that has enough resources to complete the task. We are assuming that our base station is the most powerful node before starting the work. And in the proposed model, the nodes are static, because if the nodes are not static, we have to use some mobility model. These are the symbols we are going to use in our in LTS, in our lightweight trust estimation scheme. S means successful interaction. U means unsuccessful interaction. Means two nodes are not able to communicate. Unsuccessful interaction. Rn is a random number. UID is a unique identity number. Psi is a punishment coefficient. Alpha is a reward coefficient. We have to decide some threshold value of the trust, theta. Then we are going to use a timing window consisting of 4 time units. We are checking using RID whether the data reported by the sensor node at time delta t, because time matters in trust evaluation. If you trust a person at this time, it may be not guaranteed that after 2 years or 1 year or 30 days, your trust value will be same. So trust value is calculated on the time. And we are using a feedback communication trust using F feedback data trust, and these are the some symbols. N means size of each clusters, capital N means total number of nodes in the network, and G means number of clusters. This is the flow chart of the proposed model. We start the deployment of the sensor nodes. We divide the nodes into cluster using the leech algorithm. We make the trust decision. First, we calculate the intra-cluster trust decision. Intra-cluster means we are calculating between a inside a cluster member to cluster member, and indirectly we are calculating cluster head to cluster member. So within each cluster, we are calculating two types of trust, direct and indirect. Direct cluster member to cluster member and indirect cluster head to cluster member. If this trust value is below the trust threshold, then we will raise an alarm and eliminate the node because it will be a malicious node. If not, this value will be returned to the cluster head, and then with the trust evaluation will be considered between all the cluster heads of the networks. And then again, it will be compared. Base station will calculate the trust value of all cluster head, and it will compare it value with the threshold value. If it is less than the threshold, then it is again eliminated from the network. Otherwise, the final value will be returned to the base station. Now let us discuss the trust evaluation equation that I have designed. This is the communication trust. I have used S upon S plus U. S upon S plus U means we are considering the success ratio. Uh, S plus U means total interaction, S means successful interaction. So out of total interaction, how many interaction will be successful? So this first term is successful interaction. Second term 1 upon psi to the power U. This, if the value of U will be increased, then the lower term will be increased and the punishment will be increased. So I have used the penalty coefficients to penalize the trust model, because if a node malicious, if a node behave malicious, then it will be penalized. The third term is S upon S plus 1. It is the reward parameter. So why the S upon S plus 1? Because if a node is behaving correctly today, so its reward term must be increasing slowly, not fast, because reward must be, or reliability must be increased slowly. If we increase the trust value within a 1 day or within a based on the observation of limited time of, then it may provide the wrong results, because the node is behaving today correctly may not behave correct in the last 3 hours or after 3 hours. So this is to provide the minor reward. So reward will be more and penalty will be reward will be slow and penalty will be high. So we can calculate the we can get the exact trust value, or we can get the accurate trust value. We have deployed this equation into MATLAB, and we have checked that if the success rate on the x-axis is increasing, how the trust value depending upon the value of psi 1 2 3 4, how the trust value is increasing on the y-axis. So trust value must be increased slowly, as the these lines are showing. So in which line the trust value is showing slowly, that value is optimal for our trust model. It may be changed from application to application, but for the current situation, this is showing that which value provide the slow increase in trust value. After calculating the trust value, we compare the status of a node using this equation. If trust value is between 0 and theta, we can say it is a malicious node. If more than theta and it is less than 3, because we have considered the range between 0 and 4. So if it is less than theta means it may be 2. If it is greater than 2 and less than 3, it is legitimate node. And if the trust value is greater than 3, then it is highly trusted node. This is the time window scenario in which we are considering 4 units to record the successful interaction, unsuccessful interaction. These interactions are inserted into the equation 1, and then we calculate the trust value. So in first unit of time, we have 5 plus 7 plus 6 plus 5 successful interaction, 2 plus 3 plus 1 plus 3. This is unsuccessful interaction. After a some period of time, this logical time window slide and it forget the previous interaction, because while we are calculating the trust value, it is not good practice to consider the interaction that happened 1 year ago. We have to consider recent, we have to focus on recent interaction more. Now, using the same approach, we calculate the data trust between the nodes S upon S plus U. In the same way, we provide the reward. In the same way, we provide the penalty. Again, we compare the using the same equation, whether the is a highly trustable node or malicious node. Now we check whether the data report is successful or not. If the data reported by a node on node X at delta t time and the difference between the two nodes about the data is less than the row, it is a error tolerance parameter, then we can say data is correct and it has some minor that is negligible error in data. Otherwise, if it is greater than row, then it is not correct data, it is corrupted. So we have used some data successful report data equation in our model. Now when all the cluster members have evaluated their trust, then this trust value is forwarded to the cluster head of the each cluster. So cluster head CHC, last equation shows that the trust value of the node 1 about node 1, trust value of the node 1 about node 2. So these are the values stored at the cluster head. Now all values all the clusters has reached to the cluster head. Now cluster head base station will calculate the trust value of cluster head. Now we have calculated the direct trust. Now we are calculating. We are also considering feedback trust. So feedback trust means we have to take some knowledge from the peer nodes. So we have taken the feedback from all the nodes that is assigned weight W2 from K is equal to 1 to A plus B, where A plus B, A and B are the good and bad nodes that has evaluated based on the previous equations. And this 4 into A plus 1 plus A plus B by 2, this 4 is used to keep the range between 0 and 4, and A and B are the successful and unsuccessful interaction, good and bad nodes. And this equation is derived from the Bernoulli theorem. In the same way, we are calculating the data trust from the last equation. After computing all the trust, we are calculating global feedback trust using equation 4. The 4, we are taking the seal value of this global feedback trust. The final trust we will calculate using the trust value at delta t time, then and global feedbacks. Means we are aggregating direct trust and indirect trust, and we are giving 50-50 weightage. Divide by 2 means we are giving 50-50 weightage, and the final node status we will be given from the last equation. So we have calculated direct trust, indirect trust, and within the cluster, cluster head to cluster head, cluster member to cluster head. Now this is the cluster head to cluster head communication using the same equation, but now the communication is going to happen between cluster heads, not cluster members. Similarly, all the cluster heads sending data to the base station. Now base station has arrived data from all the cluster heads, and base station will use some equations to remove the outlier using they will find the median absolute deviation, and he will remove the outlier from the cluster heads. So these are the some equations that have used in previous models, S upon S plus U. Then some authors develop S upon S plus U, S plus 1. Then LDTS developed this equation, and we have developed the last equation based on some knowledge, based on some reasoning. We have developed some, and then we have simulated this work using the MATLAB that got some good results. We have calculated the communication overhead because each node is sending data to its peer nodes, so it may increase the communication overhead. So we are going to develop lightweight communication, so we have to reduce communication overhead. These are the parameters we have used in our work size. The range of nodes we have considered 500 to 110,000. Initial energy we have considered 3 joule. And first order radio model is used in either WSN or it is called as energy model used. And radio energy used, we have taken 35 meters. Initial test value of the sensor node was 4. Range of the test value 0 to 4. Trust threshold, as I discussed, 50% I have taken. You can take more trust threshold as per the depend upon the applications. Like agar it is a defense application, so you have to use some critical trust threshold. And the range of the punishment parameter we have used 1 to 3. So these are the parameters we have used during the simulations. We have injected some malicious node 10 to 40% to analyze to check the effectiveness of our trust models. This is the simulation topology. These red dots are the malicious nodes that we have injected in our networks. We have analyzed the severity of our proposed model as the success rate of increasing, and we have compared with ADCT that I have discussed in previous table. It was an existing approach. Then I have found that our scheme LTS increase the trust value or raise the trust value slowly, but ADCT, it increase the trust value very quickly, but it is not good. Trust value must be increased slowly as the success rate increase. Because if we talk to a person and within 1 day, can I increase his value 100% that his test value? No, because whenever we will interact for weeks, months, and years, then we can say, this person is good. That's why trust value must increase slowly. So using this results, we can say that our proposed model increase the trust value slowly. So it is good than ADCT. We have calculated the values on different parameters by increasing, decreasing the value of phi and alpha, and got some good results that LTS is better than ADCT. These are the results that shows the comparative analysis of the trust value obtained of my trust model LTS and 2 base models ADCT, LDTS. This is the communication overhead incurred in my proposed model and existing models, and it shows that our communication model LTS incurs less communication overhead because we are using clusters. We are not using entire network as a whole network. We are using clusters, so we have reduced some number of communications. We have performing some communication between clusters, so some communications we have reduced. That's why my communication overhead is less. This is the worst case analysis of communication overhead with the 1000 nodes. So it is also less in my work. We have detected malicious nodes, and it shows that our model can detect 35 of malicious nodes in IoT consistent of a total of 500 nodes. So it improved 14.28% over LWTM, some base models. So it shows that our proposed model is effective. These are the findings we have discussed in previous slides. Now, our proposed model, we have designed some equations for the trust model. Now we are going to discuss how to update the trust value in our second objective. So let us discuss. This is the trust model. I have discuss. This is the equation I have discussed. This is the same equation I have discussed in previous slide. This is the same equation that all the values are going to cluster head or base stations. So we are not repeating again this content because this is the main part of this objective 2. So there are 4 cases when 2 nodes interact. First case is that all the interactions are successful if SX, Y, delta T, the interaction greater than the interaction threshold, and no unsuccessful interaction. Then we can say no unsuccessful interaction, it means trust value is fully trusted, highly trusted. Second case may be that there is no successful interaction between 2 nodes and all the interaction are unsuccessful. It means trust value is 0. Third case is there is no interaction. S is 0 and U is 0. It means when there is no interaction, we will calculate feedback trust using the last condition. And the third condition is there are some successful interactions, some unsuccessful interaction. Then how to update the trust value? In this case, we will give weightage to some weightage to new trust value and some weightage to old trust value, because old trust value must have to be considered in our trust model. If we are forgetting older history, we are not taking any values from the past history, past record of a person of an object, then we are not going to update the trust value in correct manner. So these are the four possible cases when two node may be interact, may not be interact, successful interaction may be more than unsuccessful interactions. So this is the trust update algorithm. This was our objective too, how to update the trust value. Again, we have used some values parameters. In this simulation, we have changing the network size. So you can use any values depending upon your work, but the concept is same. These are the results we have in changing the value of, because in this equation we have used phi, so we have checked the effectiveness by changing the value of phi. This is also comparative analysis, this model. Now our third objective is trust aware routing, how to perform the routing. The proposed scheme consists of unique features like direct communication trust, feedback trust, energy trust, data trust, and some high penalty and reward term also used. We have proposed a trust-aware sort trust path routing algorithm, because data is transmitting from one node to another node. So we have to find some shortest route and route maintenance algorithm, route discovery algorithms. The proposed routing scheme improves the packet delivery rate as well as network throughput. The suggested model would be capable of providing security against various internal attacks due to the consistency in trust values. So the objective 3 provides a comprehensive trust, direct trust, indirect trust, and energy trust estimation for route construction mechanism as well as route maintenance mechanism. It is a hybrid approach used in cluster IoT architecture. Communication behavior of sensor node estimate the trust score with the help of logical time window, as we have discussed in previous slides. So RCM considers the trust score, residual energy, hop counts to find a secure and energy efficient route. And RMM, route maintenance mechanism, report to source node about the construction of the new forwarding path when a shellfish node is detected in the current routing path. So these are the topology that have used. You can see that all the nodes are divided into 3 clusters. Data is going to sync node, that is the base station, and then is going to the end user. Within each cluster's node are communicating, and then cluster head are communicating using yellow lines. So using this, we have reduced communication overhead because all the nodes are not interacting. The nodes that are within the range, that are communicating easily, frequently, and it improves the trust value. It provides the facility to calculate the trust value more accurately. Again, time window, we have used some time window and record some successful interaction, unsuccessful interaction. So we have used some updated equation. We have used some beta distribution in the first term to estimate the trust score based on the communication interaction. So it will improve some accuracy of the trust model. In this equation, we have used some Q. One term is Q. Q is it provides the effects, because while you have deployed some node in like a forest area, and due to some earthquake or rain, your sensors are not able to capture correct values. In some cases, Q provides the facility to update the trust value according to the situation or some abnormalities. So Q will be calculated like abnormal attenuation factor, improve the accuracy of the proposed test model by considering the impact of external factors like network congestion, packet loss. It will be calculated using total number of unsuccessful interaction by the selfish attacks divided by total number of unsuccessful. And we use the same equation as we have discussed previous to categorize the test or to find the status of a node. Similarly, we are calculating data test. So this equation is most important part of this talk. How to calculate, what is the reasoning of the equations. In the different model, you have to find some equations using the same reasoning. Using the same logic, we have calculated the feedback test or indirect test. This is the energy test. This is the new test metric we have introduced in our third objective. If the residual energy is less than the threshold energy, it means it cannot be participated in routing because sufficient energy is very less. It can perform only basic operations. Otherwise, if it has sufficient energy, then it can participate in routing, and energy will be reduced by 4 minus j into pj, where pj is the represent the amount of energy expenditure up to jth packet, because every packet processing takes some energy, so energy is slowly decreasing. So this is the equation that we are using for energy trust. Now we have direct trust, indirect trust, and energy trust. So we are going to merging by giving some weightage to all the trust metric, and we are obtaining final trust. So we have obtained final trust, then we can find the status of a node, whether it is trustworthy or not, using the last equation. Now we have trust values, we have trust score of all the nodes. Now routing processing will be starts. So we will calculate a comprehensive path cost. First, using the hop count, using residual energy, using the trust value. So CPC consider reliability of intermediate nodes, their residual energy and hop count to find a secure as well as any energy efficient routing path. Now during the route construction phase, the protocol uses the route RRP and RREQ. RREQ means route request packet, and RRP means route reply packet. So a route request packet will be forwarded from the source node, and it will note the information of the all the route, and it will stop at the last node. Then from the last node, the route reply process will be packet to the backward, and it will reach to the source node. Then we find a path from source to destination and destination to source. So when a sensor node having enough residual energy and less trust score receive an RRQ frame, it replies that RRQ frame to precursor sensor node discarded because it has low trust score. So low trust node score, low energy nodes are not participated in this trust evaluation model. So nodes trust score of the nodes means the nodes interacting are reliably, and less energy nodes are not. It means all the nodes have sufficient energy to uh, process the data and forward the data. It means our network is secure. We have considered multiple factors, so our network achieve good throughput, good energy efficiency, and stability. This is the route discovery process, in which the input we are giving sensor nodes, energy threshold, route request, route reply, time to leave, current node kaun si hai, previous node, trust value of sensor node, trust threshold, and we are output is route discovery. So initialize we route request packet, and while route request TTL get not greater than 0, then it will go to downstream node, means it is going to the towards the destination node. If there is no destination node, then that node will become the last node. And if the trust value is less than theta, it means we have to discard route reply from the precursor node. Otherwise, start route reply process, and TTL value will be decreased by every hop count. Similarly, route reply. If RREQ TTL greater than 0, then route reply process to the route reply means from the destination to the source node. Otherwise, if the current trust value greater than the previous trust and current residual energy greater than the previous residual, and current hop count less than the, it means we have to update the routing and TTL value will be reduced. But sometimes there are some problems in the route, so we have to maintenance the route. So if energy trust of a node, suppose during this working the energy of a node is down, then in this case node X or node Y anything, unicast a RRP frame to the notify its residual energy and choose a new route. Or if trust value is less than theta, means there are two cases, either the energy will be lost or there the trust value will be lost. Then that node, it may be X or Y, send a route routing error message to the source node. If trust value is less, means there are some attack scenario in the network. So we are considering both case. Trust means we are considering attack scenario, energy means we are considering battery scenario. These are the demonstration of the how the route is constructed. This first image is the simple network. Then we have calculating the each path cost CPC. Then we are finding the route. We are updating the values, which is the near value from source node to other node, and finally from the dotted line we are getting a final route from source node to destination node using red dotted arrows. These values are calculated using the previous equations we have used. Final, this is the route that we have find from source node to destination using the bold arrows. Same we have using for this simulation work, we have used MATLAB, or you can use SN, NS simulator and other CUJA simulator for this. It depends on you, but I have used MATLAB. So we have used this position of the, because during the simulation you have to give network size, position of the base station, initial path, length, trust threshold, radio energy. So you have to give some parameter value before start of the network. So we have analyzed trust value by changing the different parameters and analyzed which parameter is optimal. Again, we have checked packet delivery ratio. Our model have good packet delivery ratio. Network throughput is also good because we have considered a comprehensive path cost. Average energy composition consumption is also good. These are the findings that we have achieved 6.2% less energy and 9.09% less energy than TESRP. It improves 3% over ESN in terms of packet delivery ratio. So overall, it provides effectiveness in throughput, energy efficiency, mitigation of attacks. So these were our three objectives we have discussed. Now the final discussion is what is the contribution of this research in welfare. So we can design a secure and sustainable sensor network welfare to enhance the security, reliability, and accuracy of the data collected by the sensors. Again, its next contribution used to monitor the air quality in urban areas using sensors. It can monitor the water quality using water related sensors. It can monitor the health, and it is being used in hospitals. It is known as IoT based healthcare, in which patient is in their home and there are some sensors on his body. Doctors from his clinic or from his own monitoring whether all the heartbeats, all the things good, well or not. If anything emergency needed, he instructed that take this medicine. So IoT based enabled healthcare is a popular research area at this time. You can use IoT sensors in agricultural productivity. Trust-aware IoT or the sensor networks can ensure the data collected in accurate and reliable, which is essential for making informed decision about crop management. This can help farmers to optimize their practice, reduce waste, and improve productivity, leading to better crop yields and increased profits. And it can also help in disaster response. means you can understand that its importance in almost all the areas where we have to calculate some trust values. Either it may be agriculture, water, air, anything. It may be food quality. So it is a very important topic, trust management. So these are the significant impact on the worlds, like the countries that are using improved efficiency. So if efficiency is improved, and positive impact on the global economy, increase worldwide safety. Hospitals and doctors are using IoT sensors, so quality of life of the patients is good and has impacted. Environment impact is also reduced and has imposed a significant impact, good impact, to mitigate the effect of climate change. It has improved worldwide health. It has improved disaster response. It has improved decision making, like in factory, in hospital, in industry, machine. If you are calculating trust value of a machine, then you have to use some more trust models. Like a car, we are going to a tour using a car. Then we calculate the trust value of a car, whether this car will go to a hill station or not. Then we go. So you calculate trust in your daily life, but you are not using some accurate trust models. You just assume that this is good. So thank you so much for your kind cooperation and understanding. And thank you so much, Dr. Roy, for this wonderful opportunity to provide me and all other faculty members uh, that is present in this talk. Thank you so much.
Okay. Thank you, sir, for this enlightening session.
Welcome,
Uh,
sir.
so now we open the house for discussion. So participants are requested to unmute yourselves to ask questions to Professor Khan. Also, you can prefer to put your messages in the chat box. So the house is open for discussion. I see some appreciation messages coming from Priyanshu Singh. Very informative
Okay,
session.
Priyanshu, thank you.
Dr. Jaya Bharti mentions very informative and interesting session.
Thank you, sir.
Okay, if there are no questions, does not seem to be the case, then please join me in once again thanking Dr. Ayub Khan for his wonderful presentation. And so I just put an e-gesture. Yeah, thank you, sir.
Thank
So
you,
I
welcome.
would now Dr. Gaurav Kumar, sir, HOD Department of Biotechnology and Bioengineering, School of Biosciences and Technology, to kindly hand over the e-memento to Dr. Khan.
Thank
So
you. Thank you, Somnath sir, for inviting me.
Thank you, Gaurav sir. Thank you so much
Thank
for
you,
this.
Tayyab sir, for
Wonderful.
accepting our invitation.
Yeah, thank you very much, sir,
and
for
delivering such an informative talk, sir.
Ji, sir. Ji, sir. It was a nice discussion with you, nice meeting with you, and hope in future we will connect again on
Yes,
some
sir.
another topic or the same topic.
Yes, sir. Thank you. Thank you so much, sir. Thank you, sir.
Thank you, sir. Welcome, sir.
Okay, then we move on further with the last speaker of today's session. I request the participants to mute, please, yourselves, so that we can conduct the session smoothly. Our next speaker is Dr. Shreya Mehrotra. Just start sharing the slide.
Shreya ma'am, are you there?
I think we are running a bit ahead of time, so maybe we wait, or maybe we can take a 10 minutes break or something, because we are much ahead of time now. Shreya ma'am is, don't see her in the list.
Somnath sir?
Yes, sir.
Yeah, if a participant want to discuss anything or want to
Yeah.
ask any question, they can unmute themselves. I have
All right.
permitted them to
Okay.
unmute themselves. So you can take some questions, sir, in the meanwhile.
All right. Sure. So the house is open for general discussion. So if participants are having some queries or something. Maybe I invite some suggestions from the participants.
जो मेरे हस्बैंड है ना वो वैसे हैं होते हैं वो ज्यादा एडवांस शांत स्वभाव टाइप है एकदम बिल्कुल शांत शांत से ये एडवांटेज
I request the participants to kindly mute themselves, please. Let me mute themselves from my side. Yeah.
Okay.
Yes,
Somnath
sir.
sir, if
Yes, sir.
participant do not have any query, we can
Mhm.
give a half an hour break and the
Okay.
session will be start from 2:15.
Yes, perfect. Okay, then participants, we request you to stay tuned until 2:15. We are taking a quick break of approximately 25 minutes from now. So we resume our session again at 2:15 p.m. and we will be welcoming Dr. Shreya Mehrotra, the last speaker of today's session. So please stay tuned and we see you again at 2:15 p.m.
अभी कर रहा है बात लंच ब्रेक चल रहा है ये सब लोग कमेंट कर रहे हैं यार मतलब भी चल रहे थे वेरी गुड सेशन कर दे रहे जो भी उसी में नीचे चैट्स में
Hello.
Yeah, hello. Hello, ma'am. Yeah, you're audible. Would you
Yeah.
like to check your slides once before we
Yeah,
start
I
the
would
session?
like to check. Yeah.
Yeah, please.
Hello.
Yeah, the slides are visible in the PPT mode. You can just make it full screen.
Yeah. So visible now?
Yeah.
Okay.
So we
Yes,
just wait
Somnath
for
sir, you
two
can
more
start.
minutes. We can start, sir. It's just two
Yes, sir.
more
Yes,
minutes
sir.
are there.
We can start.
We
It
can
will be
start.
Okay, perfect. So welcome back to our post-lunch session for this faculty development program on smart health innovation, integrating imaging, sensors, and biomaterials. So today's our last speaker is Dr. Shreya Mehrotra. So before we proceed further for the talk, I just take the opportunity again to introduce Dr. Shreya Mehrotra in front of the audience. Dr. Shreya is a leading researcher in the field of tissue engineering and regenerative medicine with an interdisciplinary background spanning biomaterials, 3D bioprinting, and microfluidic organ-on-chip technologies. She did her PhD from Indian Institute of Technology, Guwahati, followed by a postdoctoral training at IIT Kanpur. She brings a translational focus to cutting-edge biomedical research. As a Fulbright Fellow, she conducted pioneering work in 3D bioprinting at the MIT Harvard Division of Health Sciences and Technology, where she explored innovative strategies for regenerating human tissues. Currently, she is serving as a DST Inspire faculty at IIT Kanpur, where Dr. Mehrotra's research integrates advanced biofabrication and microengineering to develop physiologically relevant in vitro models for tissue modeling, drug testing, and regenerative therapies. Her contributions have garnered national and international recognition with numerous awards, high impact publications, and features in Indo-US translational research initiatives. So I welcome on the behalf of the organizing committee, Dr. Shreya Mehrotra ma'am to this faculty development program. Welcome ma'am, to the stage is yours.
Thank you so much for the kind introduction. And I would like to thank all the faculty members, especially Dr. Unvesh, for inviting me for this important lecture. And this is the next 1 hour or so, I would be trying to bring some of the
Hello.
The connection seems lost.
Shall I call her?
Yes, I think so because I think connection is lost. Mm,
She will take 1 or 2 minutes to join.
Okay.
Here it is rainy, might be the case. It is rainy here in this region, so it might be the case.
Then, yeah. Yeah, she's back. Ma'am, you need to unmute yourself.
Hello.
Yeah, you're audible now.
So is it visible?
The screen has started to share, but the slides are yet not. Yeah, now it's coming.
Okay, so basically, today I will be talking about from material design, to the functional organ, the journey, how the journey actually is built on the convergence of science, technology, and medicine, and how we can leverage some of the latest technologies such as 3D bioprinting and organ-on-chip technologies to actually develop human-like organs and have a breakthrough in the field of regenerative medicine. So before I go into these technologies, I would like to just give a bird's eye view on what these technologies are. And, we all would have heard about manufacturing because, in the recent era, the 2047 aim of India is that everything should be make in India, right? And through, one of the major thrust of the government is that we manufacture, India becomes a manufacturing hub. So when we talk about manufacturing, whether it is, your basic, commodity, whether it is your like, you have your own furniture or you have some paintings, you have carvings, you have machine parts, you have, maybe the, rockets, the space, in the space domain, you can have the airplanes, everything, from manufacturing from there to even manufacturing human organs, all of them basically, the idea started from, something which we had like materials, the basic of materials which are the most valuable commodity to actually start the manufacturing process. However, if you look at these earlier methods like carving, laser cutting, and shell casting, they were based on a very similar uh, concept that you start with a material, and whatever is not needed you actually leave out that material, and whatever is needed you make from that material you make your object, or you make your particular organ. So if you look into the manufacturing process, the process that I just explained was actually your subtractive manufacturing, where you start from a bigger material and you carve out or you make some, material or your required shape from that material, and the remaining material is left, which had a lot of drawbacks because, while you are, removing those material through various processes, there was a lot of waste. And all this waste was actually leading to a large amount of material that was not accounted for, and was also not sustainable for our economy and our for country also. So the new manufacturing processes that have been developed and are now at, really being pushed by our government includes the additive manufacturing, which is also known as 3D printing technology. And what it does is that it builds a product by layer. So there is no wastage of material, and all this material can be added in a layer by layer fashion, typically, by following or looking at a digital structure. So what it does is that, it gives you a design of freedom so imagine, like, I have put these two pictures over here, and if you see that, in the subtractive manufacturing, you would require to have a particular design, or you would require to have a particular machine that can carve out your, design, the required specified, object or the specified design. Otherwise, it's not possible. But here in the additive manufacturing, because we are doing a layer by layer method of, placing materials, there is more efficiency, and, there is a design of freedom. So because, just by layer placing, I can think of a prototype, and right from prototype, I can go onto the machine, and I can use the material, and I can develop it. So I don't need any specialized sub parts or specialized, accessories to my instrument. So this 3D printing, if I say in a scientific language, what this technology is that it is a technique whereby materials can be deposited in a layer by layer fashion, and these layer by layer fashion can then be fused to form a 3D object. So here in the image, if you can see, you can see two robotic arms that are moving. So what they're doing is they are depositing this material. As you can see, this material is in layer by layer, and it is gaining height, and all these layers are then converged together to form a 3D object. The advantages that this technology has over traditional forms of making materials, whether it is, for your implant, whether it is for your, any biological material, or even if it is any random material that we are, surrounded by, for example, even cement nowadays. So these, this technology, these materials, the most intricate structures or geometries can be mimicked with any kind of material using this technology. As I said, it is rapid prototyping right from the scratch. You have an idea, you just go to the machine, and if you have the material, you can just develop your prototype, and it is automated and highly reproducible and has high fidelity. So if you look at the, if I just give you a small idea about this technology, what is 3D printing, it's kind of started in 1980s, where, the first systems were being developed to use resins and, small, these kind of plastic materials. And from there on, we have moved really forward to 1990, the beginning of the 21st century, where, Wake Forest Institute, they first developed the 3D printed urinary bladder, and today where we have 3D printed food, buildings. So if you must be aware that now Godrej, and that's a 3D printing company, they've joined hands, and we now have 3D printed homes and flats and apartments that are being coming up. So that this technology has immense potential in every field of science, and it is very important that we kind of now adopt this technology even in medicine and in these biomedical research. So today, some of the major applications, as I said, include healthcare, aviation, automotive, food and jewelry, prosthetic, and construction fields. So in healthcare, it is being 3D printing has now, kind of been taken up, and a lot of prosthetic, surgical model, and implants are available already in the market, where they are being extensively used in the for aiding the patients that need such kind of prosthetics or implants, as well as surgeons, where the surgical models can be provided so that they can do surgeries. However, there are many areas even in the healthcare domain that we can still there is a lot of scope for using this technology, and one of them is to have these soft organs where you can actually have a 3D printed organ that is available, and you can implant them instead of using an organ transplant. Like for example, you can have skin grafts, you can have some cardiac patches, you can have some liver grafts, you can also use maybe some nowadays there are vascularized sheets that can be used and implanted in different soft organs. So all these are the new areas where 3D bioprinting is basically, or 3D printing technology is taking new strides. And if you look at the genealogy, we see that especially in the field that I'm talking about, the regenerative medicine, which kind of started in 6th century from Sushruta, where he performed the first rhinoplasty using autologous skin grafts. He was using it right from the patient's a different region, and then he's replacing trying to grow new skin. From there onwards, we have seen several milestones, whether cell cultures have techniques have advanced, or technologies have come up, or new stem cells have come up, cell technologies have newer cell technologies have come up. So different milestones we've seen along. And today in the 21st century, what we see is that, there is a need for these kind of 3D printed implants, patient-specific implants, and they are now writing the new generation of, regenerative materials and technologies. So the difference between when we say 3D printing and 3D bioprinting, what basically lies the difference is that, in your 3D printing, you basically have like a biomaterial ink, which is where you have these materials, and all these materials, you kind of just develop a 3D structure, and on top of that, you just seed the cells. So this is kind of better when you are 3D printing is when you are developing some prosthetics, or you're just developing non-living implants, like you have your bone plates, dental screws, or you have skull caps. So all these things are, good when you have 3D printing. You can just have the material, and these kind of printed structures can be available off the shelf. You can just buy, or you can purchase it during the surgery, and they can be implanted. However, 3D bioprinting is a little more complex kind of a technology. So what happens is that apart from the materials, you also have some living cells, some biological additives, or some growth factors that can be put along in these materials, and then it is printed together with the material to form a 3D structure. So once these materials are printed, they are actually living. These are like your living replicas. You can culture them, and when they mature to form a 3D tissue, you can put back the living tissue or transplant this living tissue into the human body. So this is how a conventional 3D bioprinter looks like. So, it has, because we are talking about now, not just talking about making implants and sterilizing them using gamma irradiation and ETO, but we are talking about making live implants. So this is how the printer looks like. It has a HEPA filter. It has an extruders. It allows you to print with the cells, which is it is a modular model. So it is very much, user friendly, and because of the HEPA filter and UV sterilization methods, that techniques that are being given, facilities that are given in this printer, you can easily print them with cells, and these are sterile and they can be cultured for longer durations. So generally, what happens is that, when many people would think is that, when I'm printing with cells, how can, I'm just having a material, or I have just a design, and how can just a material design, all these, things together can actually form a functional organ, right? So this journey, from the title that I gave today for the presentation, the journey from a material design to actually, developing a functional organ, basically how we achieve is that, it relies on three major things. Number one is biomimicry. So first thing is, either we choose the materials, or you use the cells such that they mimic the native tissue organization, and these materials, these cells, and the design, they come together to form a 3D structure. or it can be self-assembly where, you just give the material and the cells, and these cells, they will self-assembly. So, you just put the cells and these cells will generate cues on their own and they will assemble together to form an organ. Or it can be, either of these two technologies together. So it means that, you are not only mimicking the structure, by the design. Okay, I've given the design. If, for example, if I have a tumor tissue or if I have a liver tissue, for example, now it has a heterogeneous population of cells. It has endothelial cells, it has fibroblast cells, it has what your stellate cells. So, all of them are in a different assembly, where there is a bile duct. So, all your cholangiocytes, these endothelials, your stellate cells, they have a particular way of arrangement, right? So now if I mimic that arrangement through a design that I give to the computer, if I mimic that, then it becomes, biomimicry. And if they are able to also self-assemble, how, for example, if we put endothelial cells and if we give them some flow cues, so they actually have a way of assembling themselves into tubular structure. So, using both these techniques together, if they are able to form a small, a small building block, and many of these building blocks are put together, they can actually form a macro tissue, which is much like a human tissue. So these are basically the three major methods on which we rely that through bioprinting, human tissues can be formed. Currently, these are the three major technologies that are being used in our labs to develop human organs. For example, the micro extrusion printing. This micro extrusion printing is the beginner's level, or it is the most basic level of 3D printing. And it is very much similar to how, you are pushing a toothpaste from your tube. So just like that, so you can use a pressure, pneumatic pressure, or you can mechanically using a piston or a screw, push your material. And just as you see in the toothpaste, the material extrudes out in the form of a fiber. And these fibers are actually in 3D stacked upon each other to form a 3D structure. So, because it is very similar, it is very easy, it has robust and high cellular densities can be handled. And it is one, as I said, it is a beginner and one of the most easy techniques of 3D printing. The other is your inkjet printing. So this is very much like your desktop inkjet printers that we have, or we had been using for a really long time in the 90s. So it is just, how is in your inkjet printer. So basically in an inkjet printer, what we have is a thermal filament. So when you're, printer's ink, it glows, it passes through that thermal filament, it forms a vapor bubble. And that vapor bubble is deposited as, bubbles are deposited onto your paper, and then you see a printout, right? So very much similar to that structure, the inkjet printer actually has an actuator. And when the material passes through that actuator, it forms bubbles, or it forms these droplets, and these droplets are basically dropped. And they are dropped so close to each other that it looks like a, continuous structure, and then they can be stacked to form 3D. So basically, this is a high throughput printing technology, and it has high printing speed and high resolution, and can be used especially for drug discovery and high throughput assays. The last one is your stereolithography and the DLP, which we call digital light photography. So what it does is, it is just like how you make your photo films. So it has a bath where you have your bio ink or your material, and here you have a laser light, and as this stage moves up, the laser light focuses on the surface of this bio ink, and that is cross-linked. So basically what happens is that you are layer by layer. So for example, just 0.5 micron of layer is available for the laser, and the laser is shown on those particular layer length, and it is cross-linked. So what is happening is that, you can actually differentiate on the areas where you want the material to be cross-linked and where you don't want the material to be cross-linked. So now this is one of the most popular technique, and it is dominating the 3D printing as of now. the newer methods that are coming. However, the only problem is because it is using light to cross-link the materials, so you can only use materials that are photopolymerizable. That is, they have groups that can be polymerized in the presence of a laser light. That laser light can either be 365 nanometer, 405, or right now currently only these two wavelengths are used. So that is there. However, another important drawback of this technology is that it is very difficult to work with multiple cells, because you are using generally a bath kind of a structure. So where you can use micro extrusion, where you can have 1, 2, or 3 extruders, and each of these extruders can have different cells, and you can decide which one to use. This DLP method using multiple cells is still a challenge. So, if I talk about the most basic printing process, that is the micro extrusion printing process, it has it has 4 main steps. So the first step is your 3D scanning step. So what it does is that you would require, generally, if you are making an implant, or you want to develop a soft tissue that can replace an injured tissue, or you have had some accident prone and you don't have that tissue, you have lost some part of your, maybe a bone tissue or something like that. So if you want to replace that, so you would require basically what would you would require is basically a scan. So you can use an MRI, you can get a CT, or you can get an X-ray, and you can see from where you can get a volumetric data of your defect site or injured site. And using that volumetric data and obviously the anatomy of the human organ, you can design a structure. Once you have once you have that data, that designing of structure basically is done through using a CAD software. You can use a very beginner of CAD models, or Autodesk or AutoCAD, something that is available. Or there are many advanced CAD softwares available, which we call the medical CADs. So what they do is they actually take this volumetric data from the patient scans, that is your MRI, CT. So from the scan, they will take the volumetric data and they will develop this kind of a model. So if you see, like, this is your scan and this is the model that they have developed, right? And what it does is, once you have got these kind of a model, you just give them this, all this information is in a form of a computer code. You just give it to your bioprinter, and this bioprinter basically understands this code in the form of X, Y, and Z codes. And so it has, this kind of a extruder, which I showed, which you can fill with the bio ink. And depending on the X, Y, Z, it will deposit this bio ink along with the cells to form a 3D structure. And once this structure is formed, we go to the fourth step, that is that you cross-link them, or you can mature them, or you can, as I said, these are layered by layer structures. So these layer by layer structures have to be fused to form a 3D object. Once the 3D object is formed, you can actually culture them in a 3D in a reactor, or in your well plate, or in the most known culture conditions, which allows them to survive for long term, and you can mature them. And once they have matured, because you are just printing with cells, so they are just there, right? So if you mature them in a reactor, you culture them for long period of time, they will actually divide, they will make cell to cell ECM interactions, and they will spread out and will try to have normal tissue like morphology. And once they are developed, they have developed and matured, you can actually use them for either drug discovery, or you can use them for even for implant purposes. So this is how a basic 3D printing model actually works. And out of these 4 steps I told you, the most important step that allows that you to have a successful implant, or that makes a 3D bioprinting as a successful technology, is your material. So because today materials, by the choice of the biomaterials that we are using as a bio ink is of the foremost important property that demands, or that demands a lot of research to be done, and is responsible for the success of this technology in the regenerative medicine. So bioink being a critical component, basically we are using hydrogel-like materials, and these hydrogel-like materials basically are important because they form an interpenetrating network that has high water content and softer microenvironment. So what generally happens is that when you're printing with these hydrogel materials, your cells will not feel so much mechanical stress, and not only that, the material would allow the cells to actually have cell-cell interactions, will allow the cells to divide, will allow the cells to actually spread out and communicate with the other cells. So currently, the materials that are being used include both natural and synthetic materials, such as your collagen, keratin, chitosan, cellulose, PEG, hyaluronic acid, gelatin, elastin, fibrin, and many others. So, how to choose which of these natural or synthetic materials that can be important, or which of them, you have so many available. So, which one should be the right choice for your tissue? So, one of the major first step to, choosing the right material for developing your tissue is to understand your tissue. Now, each of these materials have their own properties, for example, your mechanical properties, and depending on, the property that would be required to develop the tissue. For example, in our human body, the heart is, like a muscle structure. It is aggressive and it is continuously beating. So, it requires to have a good mechanical strength. So it has a mechanical strength of 30 to 60 kPa. Your lungs have around 10 to 15 kPa. Your liver is much softer, has 1 to 10. And your brain, is just cellular structure. There is not much ECM because, you don't want the synapses to be disturbed. So there's hardly any ECM that is there. So it has a very little mechanical strength. It is very, very soft tissue. So it has, 1 to 4 kPa of mechanical strength. So depending on the mechanical strength of the tissue, you can actually use materials that can, mimic at least the mechanical properties of the tissue. So, for example, if I'm using something like heart, I can use silk, I can use collagen, I can use maybe elastin. For brain, I may, choose softer materials like collagen, I may use fibrin. So, all these materials basically, depending on the tissue and what is the ECM composition, what is the mechanical strength, depending on that, we can choose the basic materials. Apart from that, there are many important properties of the material that play a major role in determining the success of the bioprinting process. So apart from, being biocompatible, the material needs to have high hydration degree. That is, so that nutrient diffusion occurs when, it forms a hydrogel-like structure. It should be viscous and should have viscoelastic properties so that, when you are, for example, there is an extrusion-based method and you are pushing those materials through the needle. So what is happening? A fiber is found. So as a result, what happens is that because that fiber is being formed, so what is required is a shear thinning property. So you require cells, or you require material that can basically, when you are putting pressure, it comes out in the form of fiber and is able to deposit to form a 3D structure. Now, if this property is not there, this viscoelastic property is not there, you will not be able to. What will happen is ultimately all the stress that you are putting during the printing process would have to be borne by the cells, and you will lose on the viability of the cells. So if in case you have a material which is viscoelastic, what will happen is that instead of when you're putting a lot of pressure, it will become viscous, it will become liquid, it will slowly easily flow out from the nozzle, and it will form and it will deposit, and then it will become again solid. So this kind of this kind of sol gel kind of materials that are available are very good and aid the bioprinting process. So I have written in details, why each of them, what are the major properties that are required for these materials to actually make this bioprinting process successful. However, if you see today, not a single material can be used or is ideal for the bioprinting process. So if you look at this is what we call the paradox window. So, if you see the materials that are really, really biocompatible, they are really soft materials because, if only when, the cells, you have very soft materials, actually the cells will be able to grow, divide, mature, have maximum cell to cell interactions. But the thing is that if we use very soft materials, we actually lose out on the printability. So for example, what happens, because they are soft, they are not able to support the weight of a large numbers of 3D printed layers. And what happens is ultimately they fall down, the structure falls. So, this is completely a paradox situation. And how do we get, how do we solve this problem? So either you can, what we do is now we use sacrificial support and matrix materials. That is, we don't use one bioink, we use a combination of different bioinks or biomaterials. So for example, let me give you a very two examples. So for example, if we want to make a blood vessel, human blood vessel, what happens is, if you look at the structure of a blood vessel, it is hollow. So now if I want to develop something like this, which is hollow and also freestanding, it is typically not possible for me to do it through any kind of a traditional 3D printing process, right? Because there is no support. However, if we use sacrificial matrix and support materials, this kind of structure is possible. So what are sacrificial materials? Sacrificial materials are materials that can be sacrificed or can be removed after the printing materials. These are support and matrix materials. These support and matrix materials are also the materials that can either be there for some time and to allow the support so that the tissue can mature, and once it has matured, they can also be removed. So for example, if I want to print this kind of a freestanding blood vascular network, what I will do is that I will take a bath. This bath basically helps in printing this kind of a structure. This is a sacrificial ink. And once I have printed this block in which there is this freestanding structure, I can actually get rid of this block and I can just have the blood vessels. So this material that is in the block basically turns out to be a sacrificial material. Similarly, I can have support and matrix materials over here also, where you can print two materials together. So if I have a hollow conduit, I know that I cannot print something hollow because it will collapse. So what I can do is I can have a coaxial nozzle and coaxially print two materials. So the inner one can be your sacrificial material and your outer one can be your material with cells, and the inner material can then be sacrificed once you have matured and cross-linked this, and it will lead to formation of a hollow conduit. So these are some of the new methods that have come up and which allows to develop more complex structures, because ultimately human tissues are very, very complex. They are not like just putting bricks on top of each other, or it is not as simple as making a wall. So if you want to make human tissues, we have to think about not only the architecture but also the different cells, populations that are being there. So because of its complexity, a lot of new diversions and new techniques are being developed within this 3D printing technology. Today, 3D bioprinting technology has great potential in the field of regenerative medicine, in vitro modeling, as well as in the food industry. For regenerative medicine, it is being used for organ transplant, tissue repair, as I just said. And for in vitro modeling, it is greatly used in drug discovery, or you can use, or you want to study how a disease develops, what are the crosstalks between, maybe there is we know that there is fatty liver disease, so there is a crosstalk between gut and liver. Gut health decides how healthy your liver is, or the liver health decides how healthy your gut is. So to understand how these two organs are related, so you can actually model them. You can have a replica of these organs using 3D bioprinting, and you can study them in vitro in labs. And finally, in the food industry, this is already something that is I would say, a technology that has already been come out. It's no more, a bench-based technology. It is already in the market, and large number of, you have meat in US, even in Iran, a large number of meat companies are there, food industries are there, that they are 3D printing meat like structures, and what they're using is chicken cells, or they are using soy proteins, and they are developing an artificial muscle tissue, and which is edible. So some of the examples, of 3D printing, because I've spoken so much about the technology, that where I have used in my research is, number one, for cardiac tissue engineering. So basically we have used it as a cardiac patch for treating MI. MI basically, which we call is myocardial infarction or a heart attack. So what generally happens, I'll just tell briefly, is that there is a blockage in the artery due to deposition of cholesterol. So the artery gets narrowed and the blood is not able to flow through the artery. As a result, the heart muscle cells do not get enough nutrients and oxygen. Because they are not getting the oxygen, they are rigorously working. This exchange is not happening. They undergo necrosis, and because of that necrosis, the heart is forced to undergo into reparative measures. So what happens is that once the necrosis start, there is inflammation. The inflammatory cells enter into that area or that patch area where there is necrosis. They form a granulation tissue, and once the granulation tissue is formed, there is a formation of a scar. Now, what happens is that once the scar tissue is formed, that is, scar tissue is formed by your fibroblast cells, that part of the heart no more beats and is not able to contract like the normal tissue. It is just like how we get a scar on our hands after we get hurt. So what we tried is that using, combination of biomaterials along with different types of cells. So we basically, you have cardiac muscle cells and endothelial cells, which are prevalent over in the tissue. So using these kind of two cells, we developed a 3D printed patch, which was beating, and we seeded them with both iPSC and iPSC-derived human cardiomyocytes and rat cardiomyocytes, and we could see that this patch could actually not only look like the structure of the heart, but could also functionally also beat at around 72 to 80 beats per minute. So you can see in the video how rhythmically the patch was beating, and it provides, gives an option for using such kind of patches in future, reparative measures. So apart from that, we have also used 3D bioprinting technology for treating fatty liver disease. So, fatty liver is one of the most, which we call an epidemic right now. I think 1 in 3 of every individual has a fatty liver disease, irrespective they are fat or they are thin or they are lean. So basically, it is a metabolic dysfunction, and it generally happens due to, again, the fibrosis and due to the deposition of the oil droplets and, eating lifestyle. It's basically a lifestyle disorder with the kind of food and habits and eating and stress we have. So we are all prone to having this kind of a disease. So what we have used here is that if we are in an advanced stage of NAFLD, where moving back or, self-reparative or self-repair of the liver is not possible, so we have used these 3D bioprinted patches to actually have, so they are actually printed having exosomes. Exosomes are basically your these extracellular vesicles. So basically, they are actually the powerhouse of a large number of miRNAs and proteins that regulate a lot of key processes, especially in terms of healing, fibrosis, and lipid peroxidation. So all these are the key pathways that they regulate, and they have the miRNA and the proteins that are there in the vesicles. So these we printed these vesicles along with these biomaterials, and they now are serving as off the shelf technology. So you can just paste them using a surgical glue onto your liver, and then these exosomes are being released from these patches, and then you can see that as and when they are released, they are affecting the cellular pathways, and you can see that there is indeed a reduction in fibrosis, or a reduction even in the inflammation and the fatty liver condition of the liver. So basically, it is some of the technology, it has been well recognized right now, and we are really working towards having a sprayable also, to where these can actually be employed in the clinics now. So apart from that, you can also use for cartilage tissue. Here we have used an embedded gel-based printing technology. So this is an advancement in the extrusion technology to develop these 3D cartilages. So, because we know cartilage is kind of a paradox tissue. So you have to have a hard tissue. It's a kind of a load-bearing tissue. It requires to have a good mechanical property. but it also has some lacunae, which is a soft areas around the cell. So to have that, we embedded these fibrin fibers. So these fibrin basically provided uh, these soft lacunae like structures around these cells, and using a bath gel. So the outer layer is basically your peg kind of a material. So basically, which provides the mechanical strength of a cartilage tissue. So we could use them not only for 3D tissue, disease modeling or drug screening platform, but we could also use them for implants. We've also delved into using human bionic noses, where we've developed a nose that can be attached to some sensors, and these sensors are basically having receptors for some of these explosives, and they can be used or attached to the robots, or and can be used as bionic sensors. Apart from bioprinting, another area that I talked about in the title was organ on chip technology. So we all know that, with bioprinting, we can develop these. I talked about, we can have either implants, or we can, have tissue models or in vitro models. So, now these in vitro models, what we are doing, what we are using, how can they be used for drug screening? So what is the future of those in vitro models? So this is where that organ on chip technology comes in. So when we say organ on chip technology, last a decade or less than a decade, I would say, we all have heard the word personalization, right? We see that, now all of us, all our devices, all our communities, our connectivities, they are all, even our communication has become, everything is all about how personalized an experience we can have. So not only that, even the medicine that we are talking about has now become more personalized. Now, no more it is an era of one to one for all, but it is an era of one to one, where, for specialized things, like, for example, cancer, you have immunotherapy. Now, people talk about having immunotherapy having a large scale effect, but if you actually go to the clinics and you talk to the clinicians, you will see that, the tumors are so heterogeneous that, a same tumor in different humans may be susceptible to a particular immunotherapy regimen, while the other patient may show no progress. So, nowadays, the medicine with all these, with cancer and fatty liver as disease, all these kind of diseases becoming an epidemic, they are actually, the emergence of these kind of personalized medicines and, development of these personalized medicine is very important. And these 3D bioprinting and microfluidic based platforms, they play a very important role. So basically, these organ on chip technologies, they are multi-channel 3D chips that allows, to mimic the mechanics as well as the physiological response of an entire organ. So what I mean to say is that if you look at an organ, your organ is physiologically active. It means if you look at the heart, it is completely always, beating, it is expanding, it is contracting, your lungs are expanding, contracting, you have fluids that are flowing in, there is vasculature, there is exchange of nutrients. So they are completely in a mechanical and physiologically active state. So if we can mimic this dynamism of the human being into an in vitro system, we can not only mimic, it will be very important it will be very useful for us to actually develop human organ-like replicas. So, today, these kind of microfluidic chips, when they started, they actually were more advanced as compared to your, static 2D and 3D structures, where you were culturing these 3D organs on a well plate. So, it provided, because it is perfusion based, there is always a continuous flow of media. These kind of chips were advanced. However, today, the research has been there's so much back-to-back research that is happening in this field, that they are soon to replace the animal models, especially in the drug testing, dress testing flowchart, where, some of the tests now no more need to be performed in animals. And especially, the new NIH guidelines that have come in 2025, as well as, from the European Union, there has been a pressure to you on the 3 Rs, where you have to reduce the use of animals on in the biomedical research. So they have, so here this is where your organ on chip platforms along with your 3D printed organs have a great opportunity. So they can be used for precision medicine, they can be used for reducing animal taxing, and they can accelerate your drug development. And apart from that, they can be used for disease modeling. So if I want to just see, I want to mimic the structure disease, how a particular disease is being developed, or because of if I'm giving, for example, if I want to, if I'm say mimicking something like a alcoholic liver disease, so at what concentrations of alcohol, how this disease is being developed, what are the stages of this disease, how can be cured. So all this I can study in vitro using disease modeling. It can be used for toxicity testing, it can be used for biomedical research to study developmental immune responses, mechanisms of diseases, mechanisms of crosstalks. And it can also offer to opportunities for space studies, where you can study the effect of space environment onto these organs. So, this, I'm just giving an outline, of a very old 2013 Nature protocol, where, this is a very simple chip where they just used cells. They did not use any 3D printed or 3D structure. So what they started with is just to mimic the dynamism. So this is what they used for the lung. So they had a membrane on one side, they had the endothelial cells, which are your fluid cells, your cells of your, these blood vessels, and on the other side you have your epithelial cells, which are basically your lung cells, cells of your alveoli. And what they did is that on the side they had chambers. So on the side chamber you had basically your air. So as the air would flow, they would expand, and once the air would stop flowing, they would contract. So this was giving the expansion, the contraction, and again cues to the cells. And from the bottom chamber, they allowed the media to flow through them, so that these endothelial cells could actually behave like your, which we call, there was they could actually maintain this exchange of nutrients and would behave like a blood vessel. So this is how they mimicked, the structure. So this is how we can mimic the dynamism of the human tissue. So this is just an example. In last 1 decade, from single organs, as I just showed you, we have now been able to have multiple organ chips together, so we can have study more than one or two organs on a single platform. We can study the crosstalk, or we can study how drug is metabolized by liver, and how its metabolites are affecting different organs. So there has been a lot of innovation that is coming in. For example, in my research, we developed this kind of chip system, where we could have different strains along different these tissues, and we could study that how in case of ventricular overloading, there is high pressure, and how the heart muscles respond to high pressure, then this high stress. So you can see that on increasing the stress, we could see these mimic these different conditions. We are also using to these kind of devices, organ on chip platforms, to study inflammatory bowel disease, that is your IBD, and how we can use polymer based therapies to actually as a therapy for treating IBDs. We are also using them for personalized medicine. So what I can show you here is that we have used a platform where we can culture two cells. We've used cardiac cells and the cancer cells. So these are two different tissues that are on the same platform. You interface them with a sensing device. So what happens in a personalized platform is that you have basically so basically you have this biomimetic spatio-temporal geometry. So of these 3D printed organs, you mimic the native tissue organ, you put them on these kind of chip-like platforms, and you interface them with these sensors. So these sensors basically have so they have basically they can have your aptamers or antibodies for particular antigens that you want to sense on the action of any kind of drug, and then you can use them for personalized medicine and try to see if actually that medicine is working or not. So you can see if there is a reduction in the biomarker or there is an increase in the biomarker, depending on what kind of markers you want to assess. So we use them, here I use the similar concept for seeing chemotherapy based cardiotoxicity. So we use this kind of a setup where we had both cardiac and cancer tissue, and we were having these kind of sensors, and these sensors were then functionalized for antigens or the biomarkers that were being released by both cardiac and cancer tissue on the action of a drug. And when we were giving any kind of chemotherapy, we could see whether the condition of the heart or the cancer tissue was improving or deteriorating. So, you can see how the arrangement is done, and we use doxorubicin as a model drug. And we could see that in the absence of the doxorubicin, it was a completely good waiting tissue, whereas in the presence of dox, the cardiac tissue was destroyed. And we could observe is that there was the presently available ELISA kits, they are not able to actually detect the release of cardiac markers such as troponin T, in case of a patient that has already had a predisposed heart condition. However, it could easily be picked up by our system. So this is how this automated organ on chip technology looks like. You can have multiple organs put together on a single chip, on a single breadboard, and they can be conducted through fluid chambers, and we can actually mimic the physiological processes of a human body on a in vitro platform. We are also using this kind of technology for disease modeling and liver bone crosstalk. So as I said, you can actually use them for studying, understanding how two different organs are actually, talking to each other. What are the mechanisms that are, key to regulating the health of both these organs. So there we have tried to see how, a liver disease can actually cause osteodystrophy. So osteodystrophy is basically your osteoporosis or osteopenia-like conditions where bone mineral density is reduced, bone volume is reduced, trabecular thickness is reduced. So basically, based on that, your bone health decreases. So how a liver can influence, how your liver health can influence your bone health is what we've tried to study, and we could actually mimic them using these kind of organ on chip platforms. Hello. So yeah, so now apart from the great scope that is already there, there are still challenges with this field because real organs are really sophisticated. We are still working with improving the 3D printing technology so that we can work together with multiple cells together. As you can see, like a liver, it has more than 6-7 types of cells. So to working together with 6-7 types of cells and to get the right arrangement like the native tissue is still difficult. Tissues with dense microvascularization are also difficult to mimic because we are still working to get the vascularization concept correct. Even if we have achieved a good vascularization, how we connect a vascularized organ which is in our lab to the vasculature of the human, and how will they respond, and how will to have a coordinated systemic function, and then what happens to that 3D tissue. We don't have these kind of softwares that can analyze the performance of these transplanted organs and tissues. Similarly, with the organ on chip platforms, because they are more something that is more easy to come to the market, and translational is easier, yet they have some issues, some challenges because they need to be standardized. It is a new technology. How well they can be scaled? Can they be used for high throughput screening? What is their viability of these 3D printed organs to be used in these chips? What is their overall stability? What is the cost effectiveness? Is it cheaper to do it in animal than to actually have such integrated platforms? And because we are using human like cells, we would like to have, especially for cancer tissues, to have human cancer cells, you to have at least iPSC based cells, so it has a lot of ethical issues also associated. Nevertheless, we are moving ahead in the 3D printing market. Generally, North America and Europe are leading, but India and China are slowly coming up, and we really need to think to act fast because no more it has become 3D printing. People are going forward with 4D and 5D printing to have more complex replicas of human tissues. So, in which this is a direction that needs to be taken very soon, and there has to be a major thrust in all institutes. How it can be achieved is by having active collaborations with clinicians and surgeons. We need to understand the relevant clinic problems. We need to understand the need of the patient, the need of a doctor, how we can have a successful translation, how we can actually what is the kind of an organ that they need. We need to have collaboration between clinical engineering and pharma sectors. Even if we are developing these drug discovery, we are using these platforms, how ready the pharma industry is to take these platforms, and how we can join hands with these companies and startups and pharma giants, and actually have a translational outcome. So it can only work when we have good collaborations with scientists, and because it is a technology that is developing at the nexus of science, engineering, and medicine. We need people from all of these areas with the government support to actually have a meaningful outcome in this field of regenerative medicine. Some of the futuristic approaches that I'm very optimistic about is that, soon with the kind of discovery we are seeing, we will have an app which we called Delivery AO, which is an artificial organ delivery app, only restricted to the clinicians, where they can actually order artificial organs just like you order food using Zomato. You can have in situ bioprinting surgeries where a bioprinter can actually be attached to a human body and it can print over your defect area. um, you can have smart organs, and then finally when the organ on chip technology can be enhanced so much that you can have it on a chip where you got a full body checkup on your chip. You don't need to go to get a lot of blood, give a lot of blood for different types of tests. A small amount of blood can actually help you give results for several tests on a single chip. Thank you. So I would now be ready to take any questions if there are for the presentation.
Okay. Thank you, ma'am, Dr. Shreya Mehrotra, for this wonderful lecture encompassing different technologies in 3D bioprinting and biomaterials. So, as ma'am already mentioned, the house is now open for discussion. So, I request the participants to come up with your questions, or you may prefer to drop your questions in the chat box.
So if no one is asking, then I will ask one question, ma'am. So
Sure.
it is like, sir, like ma'am, you discussed about light mediator one, right? Where 3D printing using light laser light of 365
Yes,
something.
yes.
But as far as when we use something like a photo based, the system becomes costlier, right?
Yeah, it becomes costly. I mean, the technology right now is not very, very cost effective. As we compare to the conventional manufacturing, subtraction-based manufacturing technology. But I mean, soon it will become. I mean, it's not that we have to balance the cost and everything to the kind of product we are getting.
Because, for example, if you are probably in initiator, it comes quite costly, right?
Yeah, it is costly,
The
but also, you use a very small amount of initiative. It
okay, fine.
it is not that, I mean, it is, I think, less than, point, it is all less than 1% you use for the initiator. So, I mean, it's costly if you buy in bulk, but, and also because, it's initiator sometimes also has cytotoxicity. So you can't use a large amount of cross-linking agent or the initiator. So that's how you can balance the cost.
Okay. Actually, I don't know how much percentage people uses, but I know photo initiator they use it. For example, Jalma cross linking and other people
Yeah,
uses.
yeah.
I have seen it during my postdoc time at IIT Madras. So I found it costly, so hence I asked. So if there anyone
I
else?
yeah, I see lots of appreciation messages, and there is one small question coming from Sneha Patil. She asked, any government facility offering 3D printing? That's what she has asked.
Government facility.
Yeah, I mean, it's yeah. Her
Actually,
question reads, any government facility offering 3D printing. That's what she has
so
posted in
there
there.
is, they have, so there is BioNest, that is there. I think one of them setup is in Datta Mege. It is DHIMR, Datta Mege Institute of Medical Research in Nagpur or in Wardha. So it is in Wardha. So you can check there. So they have a BioNest, which is an incubator where, you can use these. It is a being supported by the government as well. So they have good amount of funds and they have this bioprinting facility. So you can pay the user charges and you can use them.
Okay. So would you like to follow up, Ms. Patil? I think does not seem to be the case. Okay, then, yeah, Sneha Patil, would you like to follow up on the suggestions, or do you have something more to ask? Yeah, she has mentioned. Thank you. Okay. So, any more questions? If that's not the case, then I would request Dr. Yuvanesh to kindly hand over the memento to Dr. Shreya Mehrotra.
Ma'am, we are happy, really happy to have you as one of the speaker, and it was like delightful moment to see how these things are going up all around the world. It seems you have summarized everything. It was a bulk amount of knowledge which we can able to see. And thanks for your time and accepting our invitation. So please find our token of gratitude for your time.
Thank you so much.
Mm.
Thank you. It is a great pleasure to have, being, invited to such kind of meeting, and because these are technologies where there is a lot of trust from the government. So, the more people are actually exposed to this technology, it is beneficial both ways.
And actually, I believe there is a call in DST now on adhesive manufacturing and bio manufacturing, adhesive and bio manufacturing.
Yes,
There is a
yes,
call recently, might be yesterday or
yes.
a few days before, might be. And it is interesting.
Thank
Thanks.
you. Thank you.
And Sir Somnath sir, shall we wind up?
So
I
I
think it
can
was a
log out now?
wonderful, yeah, so, sure. So it was
Okay.
really a wonderful learning experience for all of us, I believe. And then we can conclude the session today, and we again hope to see you all in the third day of FDP program tomorrow. And we have shared the link already on the chat box. So participants are requested to kindly fill up the feedback link. And then we call it a day. So stay tuned for one night, and we get you all tomorrow through the same Zoom link. So I wish you all a good evening and see you tomorrow.